So in theory, the microphone is working. In practice too, excellent. Good morning, Providence. I'm glad to say it's Friday, except it's not Friday. It just feels like a Friday. So it's Thursday, it's day 5. Welcome back. Another sunny day here. To run through a few quick things. Fire safety rules haven't changed in this state. You know where to go. Code of conduct hasn't changed. You all know it by now. We've abode by it. Everything's recorded. Speakers' dues are their own, particularly if they work for EPA, but also for everybody else. Everything will be on Hoover. And we ran plenty of tests. Everybody was negative, and of course everybody's vaccinated, so excellent, thank you. We're still gathering on the traditional lands of the Mashpee, Niantic, and Narragansett tribal nations. We honor their histories, ancestors, and perseverance in our communities today, as our country was built on indigenous land. We acknowledge the indigenous people and honor their stewardship of the land, sea, resources, and that they hold sacred and that we shepherd together. So, saying thanks again to the Brussels group that had a long day, and half of that we watched mostly live. Jared, Philippe, Tony, and Alicia, who helped run the session, troubleshoot behind the scenes, behind the back to make sure everything was running. And then the very engaging talk session in the afternoon with Angie, Jen, Katie, Ayman, and Kerry. And then, of course, as always, high output Inner Space Center, Russ Morantz, for supporting us. So in a bit, we'll have the poster winners. So it'll be 4 presentation, and Kerry and Yitka will take that over. And so what we had planned initially for this time slot was a Q&A for the toxicology session. But at the time, we didn't realize how Hoover was working, how well it is working, how well it is adopted. So basically, the questions that have come in have been answered already by Jennifer, Ayman, Kerry, and Katie. So we don't think we have the need to do this right now. If there are additional questions, use Hoover, let us know, and it seems like the ladies and the gentlemen are very happy to answer them as needed. So with that, I cover some other things that we had planned for a little later so that we can all enjoy more of the sunshine. And that'll go through 9:30, then we do the posters, and then we have the last but not least session of PFAS in the Arctic. Very exciting broad pictures, stories on PFAS and health in the Arctic. Then we do a quick closing, have lunch, and we all have survived 4.5 days of FLUOROS Global 2021. So with that, I now rely on the guys behind the scenes to make a magic switch, and it has happened.
Hi everyone, Reiner.
Sort of. So big thanks to our sponsors. That's probably a good place to start with. And it was a pleasure. This was a big trial to have a meeting that is real in COVID times, to have a global meeting, satellite meetings, hybrid. We had 45 speakers, and I didn't really count, but it felt like 45 hours of complete recording that is online. So if you ever have a long weekend and it's raining, you can go to Hoover, you can just watch it all in one big binge session. We have 21 live posts, including those that are coming up today. We have over 100 in Hoover. And we're almost done. It's not a bad thing, but for some of us, particularly Alicia and Wendy and the Inner Space Center, and it's a bit of a relief because it has been a little taxing. So the other part that we promised NIHS, and I guess because they fund us, we have to follow what we said. We promised them a white paper. We're not fully there yet, but at least I thought I can go through some of the highlights that were either shared with me or that I've put together from what we've covered over the last 4.5 days. And it started with a training session, and that was very interesting because they pulled in several state regulators from the Northeast. Gloria Puss from New Jersey, Caroline Putas was here from Rhode Island, there was Dr. Simmons from Maine, and number 4 was, Laurie Matthew from New Hampshire? Connecticut, sorry, other direction. And they said a few very interesting things that it was a training session, so not everybody was watching it probably at the time already, and people were traveling, but they really stress that communication is important for different audiences, and they always stressed effective communication. And I think that doesn't only apply to trainees, it applies to all of us. And then the other thing they all agreed on is that they're really waiting for the US EPA to step up and take leadership for PFAS regulation and rules in the US. Because they're all doing their own, but it's a patchwork and it makes it very complicated. And that same was picked up by the Australian session later on, where they basically said, look, it's so difficult to remediate if you have very different jurisdictions and different rules. So in theory, you can remediate a groundwater pollutant mass, and you might hit Rhode Island, you'll have slightly different ideas of how clean it has to be. And the other thing that was part of the first day through Andrea Amico, but of course Emily Donovan, shared the same story, is that communities are not really seen as critical stakeholders in any of these discussions. But they bear a lot of the fiscal burden, of course, the mental burden, the health burden, and the medical profession is not always there where it should be to give them guidance on what to do. And then Elsie painted a very big picture of PFAS fate transport all the way to bioaccumulation and human exposure. And I think she really honed in on one thing is the role of all these precursors. And some advances they've made really say you can come up with the main parts and try to explain all of what is an AFFF. And that's, of course, what you then need to know if you look at bioaccumulation. And the other part was, another missing piece is probably the atmospheric pathway. And we've had this through numerous sessions that the atmosphere is one of those wildcards. We never thought it was important because they are mostly ionic, but now we realize it must be important, at least at some regional scales. The second day started off with the science policy from Zurich, and it was picked up, of course, again in the human biomonitoring session, the fact that in the EU now the proposal is at 4.4 nanograms per kilogram body weight, and that really results in a drinking water guidance at around 2 parts per trillion, which is very low and would mean that most waters will have to be treated. And that is probably justifiable on the human health grounds, but also means it's very expensive. And so somebody will have to pay that. The other part that came out of it as a corollary is the fact that the tools that we have, the analytical methods, are not really geared to be at that level. The US EPA method that we have right now that are going through the approval steps have detection limits that are in the single nanograms, sometimes higher. And that's perfectly fine if you regulate at 70 parts per trillion. It may be sufficient if you regulate at 20 parts per trillion, but certainly not sufficient if you regulate at 2. So there's some more work to be done on whether this is all just a matter of bigger and better mass specs, or whether you start using passive sampling approaches. Different ideas might help figure out how to regulate at that level. The essential use discussion is certainly taking off in Europe. It's part of their big framework for how to regulate PFAS and move towards what they call the green circular economy. But even in the US, the state of Maine has passed a law that is going to outlaw any unavoidable uses of PFAS, which is the same idea. And I think the devil's in the detail. It sounds like an easy definition of what to do, but how you implement that, who has to decide, big questions, unsure. The other thing out of Europe is the idea of actually regulating a total PFAS number. And they all talked about, peaks on chromatograms. So it's going to be interesting if that's all you need to know. You just have one big run, and then you just sum up every peak and see whether that is more or less than 500 nanograms per liter. That's like the very old days of chromatography where you really couldn't resolve things. You just covered everything. Oh, yeah, that's its total. So we'll see how that's going to happen. And the last one was slightly different, but related to the fact is whether fluoropolymers are really distinctly different and can be looked at on their own, or whether they should be part of the family of PFASs. And I think we see more of those discussions and battles as we move ahead with looking at the uses of PFAS. Then we had the recorded session that happened much earlier in China. And of course, I think most of us are aware that China has become a major production ground for PFASs and fluoropolymers. And so sometimes they have a level in the environment that are shockingly high. And F53B was seen as a good replacement, but clearly the work has shown that it's just as bad as what it was replacing, which was PFOA. And so at least if you stick to the PFAS side of using processing aids that are fluorinated, then the 6 to FDSA seem to be much better. Of course, Some of us would argue that it's much better to have something that's actually non-fluorinated. But all right. And then the other part is the big advances on the non-target discovery. So we know a lot more, and certainly we've seen this in the US at some case studies, but also on the Chinese side. But those are mostly good for the anions. There's more difficulty with the neutrals, cations, with ions. And of course now there's the marriage of the new and the established using deep learning or artificial intelligence approaches to actually start data mining all these peaks. The remediation session I found very fascinating because I wasn't aware how far ahead Australia is by really using not only the good old tried granular activated carbon, but actually using surface active foam fractionation for bubbling out PFAS from water, that they're happy to thermally treat soil offsite and get it back, that they are adding carbon to contaminated, slightly contaminated soils to keep them on site. So it feels to me like they're way ahead. And it seems to be acceptable to the public, and I think at the end of the day, people just want clean drinking water and somewhat clean fish, so then however that's achieved doesn't really matter as much. They did point, of course, the problem of the conflicting regulatory targets if there's no federal or national guidelines. Lutz talk obviously point out there's a heck of a lot of different ideas of how to remediate, but a lot of those are just not ready for routine applications, which is always challenging because water treatment providers probably want something that just works and is solid and they can live with. All right, now, Amila was, of course, the overachiever. She didn't just share a slide, she shared a recording. So now, calling on Alex to make this happen. Of course, he doesn't disappoint.
Me to give a post-game commentary on how our wildlife session went on PFAS on Tuesday. So I first want to start off by acknowledging the excellent speakers we had on Tuesday. They really covered a lot of ground on wildlife measurements, biotransformation, ecotox, and bioaccumulation. So Rob gave the keynote presentation for our session on Arctic wildlife, and he presented a lot of data, and I want to just zoom in on one paper in particular because I think it's quite representative of Arctic wildlife. This is by Healy Rauti on polar bear plasma from Svalbard. It's a temporal study, and she's reporting declining PFAS concentrations over time since the late 1990s, whereas PFCAs are not showing that trend and, if anything, are increasing over the same time period. Now, I think this is quite interesting that the profile of PFCAs that we see in Arctic wildlife is quite unique. It's different from what we see in fish and birds from mid-latitudes and certainly different from humans. We do see a prevalence of these long-chain perfluorocarboxylic acids, and in particular, the odd number, C9, C11, and C13. So this is evidence of long-range transport of these contaminants to remote Arctic systems and significant accumulation in the tissues of Arctic wildlife. Now, this came up during the question period that Rob's presentation was really on these substances that can undergo long-range transport. But of course, many PFAS do not have the properties to undergo long-range transport. Should we still be concerned? And yes, of course, I think we should. Long-range transport is a criteria under the UN Stockholm Convention on Persistent Organic Pollutants because it recognizes that no single government can act in a way to mitigate the risk presented by substances that undergo long-range transport. It really takes that international cooperation. If there are PFAS that don't undergo long-range transport, they're still likely of concern. It's just the Stockholm Convention is probably not the mechanism for doing something about it. Jin Liu from McGill talked about biotransformation extensively, presented all sorts of different precursors and pathways that produce these persistent perfluorocarboxylic and sulfonic acids, as well as persistent intermediates like the fluorotelmer carboxylic acids. I really like that Jin highlighted this publicly available database, NVPath, which serves as a prediction system for different biotransformation of different contaminants, including PFAS. It's really come a long way. And also, you can add new rules to NV paths. So as new information becomes available, we're really mobilizing that knowledge in real time. Now, of course, such databases are entirely reliant on existing data. And there are a lot of PFAS for which we have no information on biotransformation. For example, Jin highlighted these two fluorotelomere beta-enes that make up our contemporary AFFF formulations in Canada. And after 120 days, they did not observe any biodegradation. The polyfluoroalkyl ethers, so the Gen X type compounds, F53B, the Solvay formulations, we don't have any information on their biodegradation beyond the fact that the OECD standard biodegradation test does not seem to indicate any biodegradation potential. So this is quite a hot topic for studying transformation processes, or potentially. Chris Solis talked about ecotoxicology in our session. He highlighted this study on multi-generational exposure of PFOA and other PFAS to these freshwater midge larvae. So over 10 generations of exposure, effects were seen in every generation, but not in a way that would be predictable with generation. There didn't seem over time to be an increase in sensitivity or an increase in desensitivity. But what was interesting is that when they took the 11th generation, so the offspring of that 10th generation, and exposed them, they found very significant effects. So what this means is that you can expose a population over multiple generations, and yet there does not seem to be an adaptive effect or a decrease in tolerance. The population remains stressed, and the effects are presented. I really enjoyed this comparison of these two different studies looking at the toxicity in bobwhite quail. The first study on the left there is by John Newstead, which was a dietary dose of PFOS, and the one on the right is a more recent study by Nicole Dennis, where drinking water was the exposure pathway for PFOS. Very different doses, so the dietary dose was at a much higher level. And yet, when they looked at the birds, the toxicity was quite a bit greater in the drinking water exposed birds. When they looked at the liver concentrations for the dietary versus the drinking water dosed birds, they found relatively high concentrations in the drinking water birds. So what this means is that we really need to track that internal dose, and that's not predictable with whatever the external dose is. And Pierre Labadie touched upon this in his presentation on bioaccumulation. This is a graph he put up from the paper by Larry Burkhardt that summarizes field and lab data on bioaccumulation with respect to the water concentration. Now, this is completely different from what we would expect. We wouldn't expect any sort of relationship between BAF and water concentration. This should be a flat line. But actually, we see this inverse relationship. And so quite a few studies are now showing that bioaccumulation declines with increasing exposure concentration. This is telling us that active transport is a factor in determining what that internal dose is. And this has really important implications for when we discuss toxicity. That's no longer sufficient to just report a nominal external dose, but we really need some insight into what that external dose is translating to with respect to the accumulation in the organism and the toxicity. So I just want to end with this this figure that Anna Roebuck put together that kind of summarizes the critical information that we have amassed over multiple decades on PFAS in wildlife, but all the questions that remain unanswered or unidentified. I'm sure there are even more questions here that we just need to figure out to better understand the effect of PFAS on wildlife. Thanks very much.
Beautiful summary. I'm so glad Amila's doing my work. No, I asked all the chairs, and Excellent. The HPMU for you, that was the human biomonitoring session. I think the key message is based on the expert analysis of the risk that really they're looking at very low safe levels of body burden. So basically, a significant part of the European population is above what is considered the safe threshold already. And if you apply the same logic to what is in the US, you're looking at tens of millions of Americans who are above that. So that's the challenge, because it also means what do you do about it? And that, of course, was raised by Philippe at his final points. You know, now we know that a lot of drinking water will be above, a lot of the breast milk is above. What do you do about it? And that's no easy answer at this point. I think the key part is also the exposure source and how you can take, how you can address and potentially regulate exposure that comes from unusual pathways, like in the case of cosmetics. So obviously the Europeans think diet is really important, sometimes drinking water, but then can you take care of all these other really exposures that really shouldn't, are not necessary and can probably much more easily curtailed. A big discussion throughout the day was or throughout the conference was what do you do with the novel PFASs? Some might be regionally important, but how do you track them? And that was the same question they argued. What do you do if you might not expect them, but shouldn't you at least screen for them? So I think that discussion will keep happening because we discover more PFASs, and it might also be that there's regional differences in what is produced in the US versus what is produced in Europe or in China. So that might also lead to very different approaches. As far as I know, F53B is much less prevalent here than it is in China, for example. Yeah, and then of course the problem that some communities are hard hit because typically when the drinking water is contaminated, it also means the environment, and that means homegrown produce and local fish is also of concern. So then you have it really from all angles. The talk session, very engaging yesterday afternoon, to make use of these PPI-era alpha mice as a novel tool for better PFOA-induced dyslipidemia. The idea of activating nuclear cell receptors and cell-based assays to get towards this large-scale screening, to see evidence for the biotransformation of the AFFF. And then, of course, this big pharma framework to start screening. Of course, that's complementary to what the EPA is doing with their hundreds of bioassays to get some better idea of toxicity. Of course, some would argue that you really can't regulate a compound at a time anymore, but that's a different question. That's a philosophical question, and it doesn't seem like the US wants to move away from this single compound at a time. For some reason, Laurel hasn't shared her summary of the last session yet. Could have to do with the fact that it hasn't even begun yet, but I'm sure it's going to be a fantastic summary. All right, big thanks to the people behind the scenes, Wendy, Alicia, Simon, Bob, Nate, who are running the desk and everything behind the scenes on Alicia's side. The Steep FLUOROS team, Inner Space Center, High Output Water Fire, who did all the technical stuff. Oh, actually, sorry, I have pictures. The poster prize team, Kerry and Yitka, who are showing up. No, not yet. Here. And of course, everybody who is here, who has sponsored us, who was online. And I assume that roughly covers us before we move on to the posters, but I'm happy to either answer questions or engage in discussion if anybody is awake enough to do that right now. But I don't want to put anybody in the spot. Okay, so we will slowly move over to the poster session in a few minutes. And then we have the Arctic session. Then I have a very quick summary of the highlights or lowlights, whichever way you want to call them, of what happened over the last few days. Then we have a final lunch, and then we can finally say FLUOROS Global 2021 is over. So thank you very much already, and I look forward to the next poster prizes and then the Arctic session. Thank you.
poster winners and have their short presentations. So firstly, we'd like to thank all of the sponsors who are up on the screen right now who supported these awards. We'd also like to give a hearty thanks to all of the judges who volunteered their time to judge the student posters, as well as all the students who, made posters and presented their posters. So big round of applause for everyone. All right, now we'll introduce our first poster winner.
Okay, so the first poster winner is actually in person, so I would like to welcome here Alexandria Forster. She's from University of South Carolina, and I believe that she will present her poster here about development a new robust total organic fluorine method to detect per and polyalkyl substances in process wastewater. So welcome, Alexandra.
Good morning. My name is Allie Forster, and I am a graduate student at the University of South Carolina, and I work for the Richardson Group. I'm super excited to have this chance to get to share a little bit more about my poster in person. This good? The microphone. Can y'all hear me? Okay, cool. So I'll just start it over. My name is Allie Forster. I'm a graduate student at the University of South Carolina, and I work in the Richardson Group. And I am super excited to get this chance to get to talk a little bit more about my poster and my work, and that has been developing a total organic fluorine method, or TOF for short, to detect per and polyfluoroalkyl substances in processed wastewater. To start, I'd like to give an overview of this project and why we're doing what we're doing. I think a common thing we've heard over and over again this week is that current analytical methods fail to capture a complete picture of organofluorine compounds with target and non-target analysis with LC-MS. However, we can use combustion ion chromatography to help gather a more complete picture of total organic fluorine, or again, TOF for short. For this project, we've really been working on developing two different extraction techniques, and that is using an activated carbon source for absorbing the organofluorines out of water. This is often referred to as TOF, or sometimes adsorbable organic fluorine, AOF for short, as well as our second technique, which is referred to often as extractable organic fluorine, utilizing a solid phase extraction. We've found that the Phenomenex cartridges, using a weak anion exchange sorbent with an activated carbon layer right underneath, has worked the best so far for us. And we are comparing these two extraction techniques for percent recovery of our PFAS standards, as well as inorganic fluoride removal. So one really important part of this project is the removal of inorganic fluoride, because ion chromatography cannot distinguish between inorganic and organic fluorine. Our current TOF method can remove up to 98% of the inorganic fluoride. Next, I'd like to move forward to talk about our actual TOF method and the parameters that I've worked to optimize. For this part, and as well as the data on the next slide, I'll really be focusing on our extraction technique that utilizes the activated carbon, not the solid phase extraction. So the first part of this method is to actually extract the organofluorine compounds out of our matrix. For this step, I've worked on optimizing our actual activated carbon source, our sample pH, sample volume, sample storage time, as well as temperature. Once we've extracted these organic compounds out of our matrix, we then combust these compounds in order to break that organic carbon to fluorine bond. This step of the method produces an effluent gas, which is then bubbled into a receiving solution. For this step, I've really worked on optimizing the actual composition, volume, and pH of this receiving solution. So now we have an aqueous solution. that contains fluoride anions that we can then analyze using ion chromatography. For this step, I worked on optimizing our eluent composition and gradient, as well as the column for this method. I'd now like to show a little bit of data that we have. On the right of the screen, I'm showing a graph of the percent recovery of our 39 PFAS standards that I've been fortunate to work with. I have used a mass balance approach for this to calculate the percent recovery. All of these standards have been analyzed alone in pure water, and currently our method has a recovery range of 64 to 112% for these standards alone. In a mixture, our current method has a recovery percent of 87% using a sample volume size of 50 milliliters. However, if we increase that sample volume size up to 500 milliliters, you can see that although the percent recovery decreases, our LOD and LOQ also decreases. We are currently still working on optimizing our LOD and LOQ and hopefully getting those lower. In the future, I'll also be working on furthering optimizing our EOF method, and again, that's using solid phase extraction, to compare to our TOF method for percent recovery. And then we will be applying both of these optimized methods to real process wastewater and river water and evaluating our optimized TOF method against high resolution LC-MS-MS. And at this point, I would just like to thank the North Carolina Department of Environmental Quality, as well as CMORS, and also my wonderful lab group and my advisor for their constant support. And thank you for this opportunity to talk more about my poster.
All right, thank you so much, Alexandria. Next we have Chuhui Zhang from North Carolina State University presenting on her poster on the stability of PFASs in solvents relevant to environmental and toxicological analysis.
Hello, everyone. My name is Chuhui Zhang. I'm a PhD candidate working with Dr. Delph Gnepi at the NC State University. My topic is the stability of per and polyfluoroalkyl substances in solvents relevant to environmental and toxicological studies. In North Carolina, both rural and urban communities are impacted by PFAS, especially recently identified fluoroethers or PFAS. For example, in our previous study, the 2015 PFAS levels in Wilmington drinking water source were 120 nanogram per liter for some of PFCAs, 45 nanogram per liter for PFSs, and 130,000 nanogram per liter for fluoroethers. These fluoroethers include mono or multi-ether carboxylic acids, as well as polyfluorinated ether acids with carboxylic or sulfonic acid head groups. This poorly understood PFAS drives researchers to study their toxicity and behavior in the environment. In our study, we wonder what is the stability of PFAS, including PFAS, in commonly used solvents, such as deionized water, methanol, acetonitrile, acetone, DMSO, and isopropyl alcohol. The workflow include we added PFAS stock solutions and evaporated to dryness. Then PFAS were redissolved in solvents of interest. The analysis took place on LC Q-TOF-MS, GC triple quadruple MS, and GC Orbitrap MS. First, we investigated the effect of solvent type. The figure is showing the stability of HFPO-DA. We can see that in water, methanol, and isopropyl alcohol, HFPO-DA had no measurable degradation in 30 days. However, in DMSO, acetonitrile, and acetone, HFPO-DA degraded rapidly, and the degradation followed first-order kinetics. Then we studied the effect of PFAS characteristics. For monoether PFAS, we had 3 linear structures and 3 branched structures. This figure showed the stability of monoether PFAS in acetonitrile, and we can see the linear PFMOAA, PFMOPRA, and PFMOBA had no measurable degradation. However, the branched PMPA, PEPA, and HFPODA degrade rapidly in acetonitrile. Then, for the HFPO homologs, we studied the stability of HFPO dimer acid, trimer acid, and tetramer acid, as well as GenX, which is the ammonium salt of HFPODA, and they also degrade rapidly in acetonitrile. Then we studied the effect of water to organic solvent ratio. The figure shows the stability of HFPO dimer acid in acetonitrile. For example, in 80 to 20% of acetonitrile to water, there's no measurable degradation. However, in 100% of acetonitrile, HFPODA degraded rapidly. Then we looked at the effect of temperature, and this figure shows that HFPODA degradation rate increased with increasing temperature in acetonitrile. Lastly, we used the GC Orbitrap high-res MS to identify transformation products for HFPO dimer, trimer, and tetramer acids. They all degraded via decarboxylation and formed fluoroethers E1, E2, and E3 respectively. Here is an example of identifying HFPO tetramer acid degradation product. So the top row is the fluoroether E3 standard, and the bottom row is the degradation product of HFPO tetramer acid in acetone. By comparing the matching retention time from the GC Orbitrap total ion chromatogram as well as the mass spectra, we confirmed that HFPO tetramer acid degraded into fluoroether E3. The conclusions are solvent choice is important when preparing dosing solutions and samples. EMSO, acetonitrile, and acetone are not suitable for storing GenX and other fluoroethers, but may be suitable if sufficient water is present. Thank you.
Thank you for a really nice presentation. I would like to introduce our next speaker, who is Jessica Clothier from York University, and she will give us a short presentation about source attribution of PFAS in wastewater treatment plant effluent.
Hello, everybody. My name is Jessica Clothier, and I'm a PhD student at York University. So today I'm going to be briefly discussing my poster on source attribution of PFAS in wastewater treatment plant influent. So if you're interested in what I'm talking about today, then please check out my poster, number 49, for more information. So current studies of wastewater treatment plants primarily focus on the effluent, and this is to assess removal efficiency as well as look at the contaminants that are re-entering the environment. However, my research is primarily focusing on the influent in order to determine some trends and sources of PFAS in response to the regulation changes of emission and usage of PFAS. And so I am looking at perfluoroalkyl acids, or PFAS. And so my data set is composed of 14 different wastewater treatment plants across Canada that were sampled in 2019. And so the total range of PFAS in the influent was between 11 and 440 nanograms per liter and 20 to 240 nanograms per liter in the effluent. And so something of note was I found no significant correlation in the PFAS concentrations of the influent with population size. And so the figure on the right you can see is a compositional breakdown of PFAS across the wastewater treatment plants, as well as some drinking water sources, which include lake water, tap water, and rainwater. And so this can be useful to determine any trends, compositional trends in the data. And so something that we can note is that short chain PFCAs actually dominate in the wastewater treatment plant influent and drinking water sources. And so we can see this in the PFBA in blue and PFPEA in purple. And so other trends that we can see is that some of the wastewater treatment plants are actually very similar in composition as well as concentration to the drinking water sources. And so this could be a possibility of recirculation of the PFAS at some of the location. And this is contrasted by some of the wastewater treatment plants that have much higher concentrations of PFAS, as well as some different compositional breakdowns. And so I also looked at correlating the different PFAS influent with each other, and this was broken into 4 different regions in order to kind of break down different sources of PFAS. And so what we can see is in region 3, and on the top figure it's primarily correlated in the PFCAs. And in region 1 on the bottom figure, we can see that there isn't that correlation that's occurring. And so this indicates that it could be fluorotelomere chemistry as the major source of PFAS in region 3. And so this is contrasted when we compare region 3 to region 4. So region 4 on the bottom here has PFSAs correlated, which we don't see in region 3. And so this could indicate that there's some evidence of the source of PFAS being aqueous film forming foams, or AFFF in region 4. That is not present in region 3. So in conclusion, changing legislation has shifted the manufacturing of PFAS to an increased use of short chain PFAS. And so also analyzing wastewater treatment plant influent can lead to some insight about the sources of PFAS. And so if you're interested, please check out my poster. Again, it's number 49 for more information. And I also did more data analysis that I wasn't able to show here due to time. Thank you so much.
Mask on, mask off. So thank you so much, Jessica, for your presentation. And our last poster winner is Heidi Pickard from Harvard University, who will be presenting on accumulation of PFAS precursors in recreational freshwater fish species, and she also sent us a video.
Hi, everyone. I'm actually going to do this virtually live. Thank you, Carrie, for that introduction. As she said, I'm a third-year PhD student at Harvard University, as well as a STEEP trainee. It's an honor to be one of the awardees, as there were many really great posters, so a big congratulations to everyone and all the really cool research they're doing. I'm just going to share my screen here, and hopefully everyone can see that. So this poster presentation focuses on the accumulation of PFAS precursors in recreational freshwater fish species from the state of New Hampshire, which I identify using a toolbox of analytical methods. And if you have any comments or questions about this poster presentation, feel free to just message me on Whova or send me an email. To start off, as you all know, PFAS are known to bioaccumulate in food webs, and seafood consumption is an important dietary exposure source, especially for some of the legacy PFAS, such as PFOS. However, there are large quantities of PFAS precursors in the environment, some of which have shown an enhanced propensity for food web uptake. Therefore, it becomes important to consider PFAS precursors when assessing environmental exposures to this class of compounds. The most common standard method for detecting PFAS in samples is targeted LC-MS analysis. However, this method only captures a small fraction of the PFAS in the environment. And so this is why other methods are needed to close that PFAS mass budget, like TOP and EOF, that can capture a larger fraction of PFAS that might be present. For this project, I chose to optimize the total oxidizable precursor assay, or TOP assay, for fish muscle tissues, in which PFAS precursors oxidize to the corresponding PFCAs of related perfluorinated carbon chain length. These precursors can be categorized based on manufacturing origin as either ECF-based precursors, so electrochemical fluorination, or FT-based precursors, so fluorotelomerization processes. And this TOP assay, in combination with statistical Bayesian inference approaches developed by my colleague Bridger Rule, provides a basis for estimating the concentration and chemical structure of PFAS precursors based on perfluorinated carbon chain length and that manufacturing origin. And so this means using these methods, we can infer what some of the most probable unknown precursors are, and then use non-target analysis to confirm the presence of these, allowing us to further close that PFAS mass budget in biological tissues. In this project, 8 species of recreational fish were collected from 9 freshwater ecosystems in southern New Hampshire, an area with both known and unknown sources of potential PFAS contamination. The targeted analysis of both fish and water samples showed differing compositions in analytes detected. In the water shown on the right of each of these composition plots, the shorter chain PFCA shown in the light pink were detected with the greatest frequency, whereas in the fish shown on the left, PFOS shown in blue, the long chain PFCA shown in the darker red, and some of those ECF precursors in the green were detected in a greater frequency, suggesting that the long chain PFAS and precursors are likely bioaccumulative since they're not detected above the limits in water. In order to assess the bioaccumulation potential, we calculated measured BIFs, so bioaccumulation factors, for the C7 to C10 PFCAs that had detectable levels in both the fish and water, which showed increasing BIF with chain length, as has been shown in other studies. To assess the bioaccumulative potential for those ECF precursors, minimum estimated BIFs were calculated using the water MDLs, which showed that these precursors have minimum log BIFs greater than 3, indicating that they possess the tendency to be really bioaccumulative. And since we only analyzed a few of these targeted precursors, there's likely other precursors present in these fish that are just as bioaccumulative. To determine if that's the case, I optimized the TOP assay for fish muscle and applied it to a subset of fish samples. Here I'm showing an example of one of those fish where after oxidizing the tissue extract, there were increases in the C4, C5, and C7 PFCA, with those few targeted ECF precursors only accounting for a small fraction of the C4 and some of the C5, suggesting that there are other unidentified precursors present in that particular sample. So using Bayesian inference, I used information from these measurements and on yields of other precursors from the literature to infer concentrations of oxidizable precursors. Here, the C4 ECF, C6 ECF, 5,3, and 7,3 fluorotelomers have the greatest probability density estimates at the highest concentrations. And then non-target analysis confirmed the presence of a C4 ECF, FBSA, and a 7,3 FT, the 7,3 FTCA, in addition to other shorter chain ECF precursors, basically concluding that this toolbox of analytical methods successfully allowed us to both qualitatively and quantitatively identify additional bioaccumulative PFAS in fish muscle. This work therefore strengthens the need for additional methods to close that PFAS mass budget in fish tissue samples in order to better assess the full extent of PFAS exposures to fish consumers. Thank you so much for this opportunity to tell you a little bit about this work.
Thank you, Heidi, for your presentation, and congratulations to you and all other awardees. So we can close this poster presentation by applause here. Thank you. And now it's my pleasure and big honor to introduce to you a chair of our last panel at FLUOROS Global 2021, Dr. Laura Scheider. Laura is a research scientist at Silent Spring Institute, where she leads the Institute's Cape Cod water quality research on PFAS and other contaminants of emerging concerns. Her areas of expertise include environmental chemistry, environmental engineering, and exposure assessment. She's a leader in characterizing sources and exposures related to PFAS, pharmaceuticals, and other unregulated emerging contaminants. Her current research focuses on PFAS in drinking water and consumer products, septic systems, and sources as sources of unregulated drinking water contaminants. Her research aims to identify drinking water supplies that are impacted by emerging contaminants from septic systems and other sources, and to work with communities in identifying and addressing local drinking water contamination. Laurel is a technical advisor to ATSDR's Community Assistant Panel at the Peace Trade Park, a site of PFAS drinking water contamination. Before joining Silent Spring, she was a research associate at the Harvard T.A. Chen School of Public Health, where she studied heavy metal contamination at an abandoned mining site in rural Oklahoma, and led a community-based research on mercury exposed among Oklahoma fishermen, including members of local Native American tribes. Dr. Scheider earned her master and PhD in environmental engineering at the University of California, Berkeley, and a bachelor degree in environmental engineering science from MIT. It is with a great honor to welcome here our colleague and friend, Laura Scheider. Welcome.
Thanks, Yitka. I didn't expect you to read my entire bio. Great, thanks all of you for being here. I've learned so much from all the speakers, all the presentations, posters, and discussions over the past few days. So it's really an honor to be here as the chair for this final session this morning. Our session today will focus on global transport of PFAS, what that means for Arctic ecosystems and for the people that live there. I think it's a perfect way to bring this conference to a close, not only because it's focused on global transport, and this conference has been so global in its nature, but also it really brings together the chemical fate and transport processes, bioaccumulation, exposure, and health effects research. So we have 3 great presentations lined up. The first presentation will be given by Cora Young at York University in Toronto in Canada. She will talk about atmospheric and marine transport of PFAS, PFAS levels in the ocean, chemical forensics, and PFAS accumulation in wildlife. Then we'll hear from Dr. John Odland from the Norwegian University of Science and Technology. He will talk about recent reports from the Arctic Monitoring and Assessment Program on human health, including information about levels of PFAS in PFAS blood levels in communities throughout the Arctic and related health effects. And then I'm really pleased that this conference is going to end where it began, featuring voices of indigenous communities and PFAS-impacted communities, which I think is such an important motivation for all of our work. Our final speaker will be Vi Waihi. She's a member of an Alaskan indigenous community, and she also works for the Alaska Community Action on Toxics organization. She will talk about her work on persistent organic pollutants, focusing on PFAS in traditional foods, and what that means for members of her community. So without further ado, I'm going to introduce our first speaker. Dr. Cora Young is an associate professor and the Rogers Chair in Chemistry at York University in Toronto, in Canada. Her research focuses on the development and use of state-of-the-science analytical techniques to probe chemical mechanisms relevant to pollutant fate and transport, air quality, both indoors and outdoors, and climate change. So with that, we'll start with Dr. Young's presentation.
Good morning, everyone. My name is Cora Young. I'm an associate professor and Rogers Chair in Chemistry at York University in Toronto, Canada. And I'll be talking today about the global distribution of PFAS and understanding its mechanisms for long-range transport. So for those of us who are used to measuring PFAS, we know how much of a challenge it is to actually find a sample that doesn't have PFAS to use as a blank. And that's because we find PFAS ubiquitously in the environment, including in remote regions far from where these products are used or created. So what that tells us is that PFAS actually undergoes long-range transport. We find it in virtually every sample we look at. And one of the focuses of this session will be why we care about this, what are the impacts of the transport, and I will leave it to the speakers in the session to talk about it in detail, but I just want to bring a few things to our attention to motivate the mechanisms that I'll be talking about for the next few minutes. So transport to remote regions far from use is an environmental justice issue. The people that live there that have not used these materials and some entire countries where they have not been using the materials become contaminated. And that's offloading responsibility on those people to deal with this pollution. One remote region, the Arctic, has a very long food chain, which leads to high levels of contaminants in top predators. And this is an additional environmental justice concern when we realize that these represent many of the traditional foods of indigenous peoples in these regions, which leads to additional exposures. And one final thing I want us to keep in mind is that when we have global contamination of chemicals, that means we need global cooperation for effective regulation. Locally regulating things may not be as effective if we want to reduce contamination to remote regions. All right, so if we want to start back at the beginning, how do contaminants end up globally distributed? Well, they have to move through an environmental media that moves. So we have air and we have water. When we think about air, air of course moves relatively rapidly, faster in the east-west direction than in the north-south direction, but generally quite quickly. Contaminants that are transported in air can become globally distributed very quickly, on the order of weeks to months. When we look at water, we also have the potential for global contamination through the series of currents in the ocean. Now, these pathways are much, much slower. They typically lead to transport, sorry, that should say water. Transport through water is slower on the order of years to decades. So we have these two transport pathways that can lead to global distribution. Now, when we think about PFAS, of course, we've been talking about PFAS all week, and we know that PFAS as a class is comprised of thousands of individual molecules that have numerous structural features. The features that are going to be important for my presentation are really those that are neutral and those that are ionizable. And those that are ionizable are effectively the perfluoroalkyl acids, or PFAS, which is what I'll refer to them as from now on. Okay, so let's start with talking about the neutral compounds, which are the easiest ones to understand. So our neutral PFAS are volatile, and so we expect them to actually be emitted into the atmosphere. So emitted through industrial processes as well as use of commercial products. And because they're volatile, they can be transported through the atmosphere and become more or less globally distributed through that pathway. And I'm showing you here some data from the high Arctic, from Alert and northern Greenland. These are fluorotelomere alcohols that are routinely found in air samples collected there. So these volatile neutral compounds are found globally in the atmosphere, not too surprising, including in remote regions. Now, when it comes to the ionizable or the perfluoroalkyl acids, the transport becomes a little bit more complicated. So these compounds favor the condensed phase. When they're industrially produced or used, we expect the majority of emissions to be into the condensed phase. Some emissions do end up in the atmosphere, but as the name suggests, of course, these are strong acids. The pKa of the carboxylates is about 0, and the sulfonates have a pK of around -8. So we don't have a huge amount of knowledge about PFAS directly, at least not experimental knowledge, but we know a lot about other strong volatile acids that we find in the atmosphere, such as nitric acid and hydrochloric acid. These are known to deposit to the ground or aerosol surfaces very rapidly, on the order of hours to a day. And so what this tells us is that the PFAS that are emitted into the atmosphere will be deposited relatively close to the site of emission. And we do have some data from nearby fluorochemical processing plants or fluorochemical production plants where we see this is the case. And we find these PFAS in water, and it's not too surprising then that we find them widely in ocean water. They're actually found globally, including in remote oceans. This is a summary of several studies published in the recent review by Derek Muir. All right, so if that were the whole story, then it might be relatively simple, but that is not the whole story, because these PFAS can also be environmentally formed. So these neutral PFAS can actually be transformed in the environment to form the acids. So this can happen through atmospheric oxidation. And I'm showing you the 8,2-fluorotelomere alcohol. It can be oxidized in the atmosphere. So that typically means reaction with hydroxyl radicals to give PFNA and PFOA. So with PFNA and PFOA, as well as the homologous shorter chain PFCAs. We can also have the atmospheric oxidation of sulfonamides. So when these react in the atmosphere, we form PFAS as well as PFOA and the homologous series of shorter PFCAs. When it comes to metabolism, we can also transform these neutrals into acids. So for the fluorotelomere alcohols, we can turn them into carboxylic acids, notably PFOA. And for the sulfonamides, they can also be metabolized, but only into sulfonic acids. So, but what this effectively does is complicate our understanding of long-range transport for these PFAS, because we have two different transport pathways. We can have direct oceanic transport of PFAS, and we can have indirect transport. So atmospheric transport of neutrals, and then transformation by either atmospheric oxidation or metabolism to form PFAS. So it's useful for us to be able to distinguish between mechanisms if we want to regulate or otherwise predict the contamination that we'll find in remote regions. So how are we going to distinguish between these two mechanisms? Well, we have several approaches that have been used. We have tracer homologs, location of samples, relationship to tracers of environmental processes. We have isomers of the PFAS, homolog ratios, as well as models. So I'm going to talk today briefly about each of these and what types of information we can gain from these environmental forensic techniques. All right, so the first technique is tracer homologs. So some of our PFA homologs can only be subject to direct or indirect transport. So these compounds can effectively act as tracers for their respective transport pathways. So if we look at direct transport, compounds that can act as tracers for direct transport have no known precursors. So some examples would be Genex or the cyclic sulfonic acid PFEX. Compounds that can act as tracers for indirect transport are those that have not been known to be produced directly. So there's basically no direct source of these chemicals. They have to be formed indirectly. And so some examples of indirectly formed tracers are perfluorocarboxylic acids that have 14 or more carbons. So let's look first at a tracer for direct transport. So Genex in the ocean, this is a recent paper that detailed a spatial study of ocean samples, and they were following ocean currents to try and understand ocean transport. So this study saw Genex in all of the ocean samples, virtually all the ocean samples, including Arctic seawater, and you can see it there in the gold color on the right-hand side. And the spatial trend they observed was consistent with transport via ocean currents, not surprising since we consider this to be a tracer of direct transport. And this indicates that direct transport by ocean currents is a feasible feasible long-range transport pathway. All right, so let's look now at a tracer for indirect transport, and that is these long-chain PFCAs. And these have been detected in numerous species in the Arctic. I'm showing you here now an example of seabirds, the northern fulmars, that were collected from Prince Leopold Island in Nunavut, Canada. And what we see here is concentrations, detectable concentrations of the C14 and C15 PFCAs going all the way back to 1975. And if we cross the Atlantic Ocean, now looking at seabirds from Norway, and again, seeing C14 and C16 PFCAs in these species. So this clearly tells us that indirect transport is occurring and is a likely source of the contamination of these chemicals, at least. So this is one technique we can use to understand transport. Now, that's useful, but if we have chemicals for which both mechanisms could be possible, we need to look at some different techniques. So the second technique we can use is to examine samples from specific locations. So we know that measurement of PFAS in remote locations that cannot be reached by the ocean should be subject to indirect transport. So we have quite a bit of data from a couple of these types of samples. So the first is landlocked lakes. And I'm showing you here the PFAS in mussel from Arctic char that was collected from Lake Hazen, which is a landlocked lake on Ellesmere Island in Nunavut. And we see, of course, detectable levels of PFAS in the tissue of this species that lives in a landlocked lake. The second type of sample we can look at are high altitude glaciers or ice caps, showing you here data from the Devon ice cap. The Devon ice cap is on Devon Island, an uninhabited island in Nunavut, Canada, and it's about 2 kilometers above sea level. So again, we see PFAS in this sample, which suggests that indirect transport is occurring. But perhaps there's a caveat that we have to consider before we can be certain. And this comes from some recent work, laboratory studies, suggesting that marine aerosols may contribute to our distribution of PFAS. So just to back up, marine aerosols are formed from wave action. So waves generate bubbles. These bubbles effectively rise through the ocean, scrubbing surfactants and forming the sea surface microlayer and a whole lot of small aerosols when these bubbles burst. And these small aerosols are enriched in organics and can be transported long distances. So this effectively takes the contamination in the ocean and brings it into the air. And as we know, in the air, things can move greater distances much, much faster. So this can be quite important. And there's been a few really great lab studies done over the past few years showing the potential importance of this pathway. So I'm showing you here data from a laboratory study. On the y-axis is the enrichment factor of the aerosols. So this is the enrichment factor in the aerosols relative to the water from which they were formed as a function of aerosol diameter. And you can see from that y-axis that the enrichment factors are up to almost 10,000. So that means we could have a really, this could be a really important transport source for PFAS. All right, so how can we understand whether this might be impacting some of our inland landlocked locations? So one way to do this is to look at sodium. This is a commonly used tracer of marine aerosols. It's very commonly used in the atmospheric chemistry community. So if we see strong correlations between PFAS concentrations and sodium, this could indicate that there's an ocean contribution via marine aerosols. So effectively a direct source. So if we go back to the samples from the Devon ice cap, we saw that there were no correlations between PFAA deposition and sodium. So no correlations That suggests that the marine aerosols are not a significant contributor to PFAS in this location. So that then confirms that indirect transport is occurring to these inland remote regions. Okay, so let's go through what we have learned so far. We have our two mechanisms. We have, for direct, we have homologs that have no precursors, have to be subject to direct transport. For indirect transport, we have homologs that are not directly produced. And for indirect, we have PFAS that are found in remote inland environments. So that's great if we're only looking at specific homologs that can be easily identified as being only direct or only indirect, or if we're looking at a location where only one mechanism can be active. But what about situations where that's not true? So what about homologs that can be formed from both processes in locations that could be affected by both mechanisms? We're going to need some different techniques to try and tease apart the importance of the different mechanisms here. So one of the approaches we can use is looking at isomers. So industrial synthesis of PFAS is done using two different techniques: electrochemical fluorination and telomerization. Electrochemical fluorination, or ECF, leads to the production of branched isomers. It's a radical-based process, so we get some linear, it's still mostly linear, 70 to 80%, and a whole lot of branched isomers, so 20 to 30%. If we look at telomerization, we end up with primarily linear PFAS produced at the end of this synthesis. So our ECF PFAS is a mixture of linear and branched, and our telomerization is purely linear. So how does this relate to our indirect formation of PFAS? So our fluorotelomere alcohols, as the name suggests, are produced by telomerization. And so the acids formed from their oxidation or metabolism are going to be linear isomers. If we're looking at sulfonamides, the acids formed from degradation of these compounds are going to be mixed isomers because these sulfonamides themselves are produced exclusively by ECF. So we can actually use isomers to identify sources. So if we look at ECF and telomere, both have possible direct and indirect contributions. But because of the production histories and volumes, the remote samples in the early to mid 2000s, particularly in the Arctic, that contained ECF branched isomers could be attributed to direct transport. So let's take a look at some Arctic samples. I'm showing you here the results of two studies, one looking at polar bear liver samples and one looking at ocean water. So there were polar bear samples collected from two locations. And in Greenland, there was about 4% branched PFOA. And in Canada, in Hudson's Bay, there was no branched PFOA. So this suggests that the PFOA that the Canadian bears were exposed to was exclusively formed indirectly, whereas the PFOA that the Greenland bears were exposed to was a mixture of direct and indirect. Now, if we look at the ocean samples, branched PFOA was observed in all of the ocean samples, and it increased from west to east. So this suggests that all of the samples were a mixture of direct and indirect, but that the fraction of directly transported PFOA increased in the east. They also noticed that there was an increase with salinity, so that there was a more direct transport, the more salty the water was. So this suggests that the fresh water, which is primarily draining from continental regions, is enriched in indirectly transported PFOA. So this suggests a greater contribution of direct transport in the eastern Arctic and a greater contribution of indirect transport in the western Arctic and also near continental sources. So another tool that we can use to understand PFAS transport is looking at homolog ratios. And so these homolog ratios are a neat fingerprint that is unique to atmospheric oxidation. So the products of atmospheric oxidation of neutral PFAS lead to homolog ratios that are indicative of this process. So the main neutral PFAS compounds that form PFCAs through atmospheric oxidation are the fluorotelomere compounds, and I've shown a series of the 4,2 fluorotelomere compounds at the bottom. So atmospheric oxidation of fluorotelomeres leads to the formation of a perfluorinated aldehyde, which I've shown on the right-hand side. And there's no influence of the perfluorinated chain length on the kinetics or mechanism. So what I'm showing you will apply to all of the different fluorotelomere compounds. So there are two mechanisms that form PFCAs from perfluorinated aldehydes under the low NOx conditions that are characteristic of remote regions. And so don't worry much about these mechanisms. I just like to keep them in my presentation because I love them so much. But the take-home message from these mechanisms is that we start with the C5 aldehyde in both cases. And from one mechanism, we get a PFCA with the same number of carbons. And from the second mechanism, we get a PFCA that has one fewer carbon. And this ultimately generates lesser amounts of the homologous series of shorter PFCAs as well. And so the yields of this N and N-1 acids are approximately equal. So if atmospheric oxidation is an important source, we expect approximately equal amounts of these pairs of PFCAs. So from that 4,2 fluorotelomere compound, we would expect to form approximately equal amounts of the C4 and C5 PFCAs. And we would expect similar from other fluorotelomere compounds. And we examined this in a 15-meter ice core that we collected from the Devon ice cap, which we met earlier on. Okay, so if we take a look at the homolog ratios through time, this 15-meter ice core represented deposition from the late 1970s up until 2014. And so starting with the 8,2 fluorotelomere alcohol, we would expect this to generate approximately equal amounts of the C8 and C9 PFCAs, and that is what we see. And if we look at the 6,2 fluorotelomere compounds, we'd expect to find approximately equal amounts of C6 and C7, which is close to what we see. And same with the 10,2 fluorotelomere compounds, we expect to see approximately equal amounts of C10 and C11. So this suggests that the fluorotelomere atmospheric oxidation is the dominant source of these PFCAs. And we already suspected that indirect transport was very important for this type of location. And this really just is a nail in the coffin for the importance of these processes in these landlocked regions that are remote from the ocean. All right, and the last thing I'm going to talk about is the use of models to predict the importance of different transport pathways. So there's been several different models that have been used of varying complexity, but models have been used to examine and predict the distribution from the transport processes we've talked about today. So direct transport, an example of a model that's been used to examine that is shown here. This is a recent oceanic model looking at PFAS that incorporates all of the different currents that are expected to be found in the ocean. This has been validated against oceanic measurements, and therefore we can consider it to be relatively robust, and therefore it can be used to predict oceanic concentrations into the future, knowing that directly emitted PFAS isn't really coming from North America anymore. We can see that through time, the concentrations of PFAS in the Atlantic Ocean become more homogeneous. So a model has also been applied to predict marine aerosol direct transport. And this model predicted PFOA emission in marine aerosols, which you can see on the left-hand side. You can see this varies dramatically. The most important factor for this is wind speed. So that's why there's such a huge emission in the Southern Ocean. And you can see that deposition to land on the right-hand side is most important close to the ocean, as one might expect, and as we would expect also based on some of the results of the tracer work that we talked about earlier. And lastly, models have also been used to predict indirect transport. So I'm showing you here the output of a geochem model that incorporates all of the complex atmospheric oxidation mechanisms that I showed you earlier, and uses these as well as complete atmospheric dynamics to predict atmospheric deposition of PFAS. And this model has been validated against environmental measurements, suggesting that it is quite robust and could be used to predict deposition going forward. One thing that we are generally lacking as a community is a model that's capable of understanding all of these processes in equal detail and thereby comparing the importance for different homologs. Okay, so this brings me to my take-home messages. So neutral volatile PFAS are globally distributed through the air, not too surprising given their physical chemical properties. When it comes to ionizable PFAS or the perfluoroalkyl acids, the transport is much more complex. There are two main transport mechanisms that result in the global distribution of these species. There's direct transport, that's oceanic transport of PFAS, which can include transport by marine aerosols. And there's indirect transport, which is the atmospheric transport of neutral compounds that are subsequently transformed to PFAS. We discussed several techniques, including location, homolog tracers and ratios, isomers and models that can be used to distinguish between the two mechanisms. But the relative importance of the mechanisms really depends on the homologs and the locations being considered. So the relative importance is going to differ dramatically depending on those two factors. And so I just wanted to finish off with some open questions in PFAS long-range transport. So one major open question that I see is sources. So it's quite clear for some homologs and some locations, while for others, a lot of uncertainty remains. And this is something that we really need to continue exploring, in particular because this impacts the effect of policy on limiting the contamination in these remote regions. The role of aerosols in transport of PFAS is a big open question. We talked about some lab work that's been done to understand the role of marine aerosols, which is fantastic. But this needs to continue to really understand the role of marine aerosols in the real environment. And the role of other types of aerosols is so far unclear. And one of the challenges we face in the PFAS community to understand atmospheric processes in the real environment is limitations of our measurement techniques. So processes in the atmosphere are very, very fast, and therefore we need fast measurement techniques in order to effectively understand them. If we're using time-integrated techniques, integrating days, weeks, even months of concentrations, it's going to be very difficult to understand these fast processes that drive atmospheric transport. And lastly, being able to quantify the relative importance of different transport mechanisms. So we really need to have environmental models that comprehensively examine multiple mechanisms. So we have really good models for individual compartments of the environment, but bringing that together, I think is really useful for comprehensively understanding long-range transport of PFAS. And lastly, I would just like to acknowledge my group and the funding for the small amount of my own research that I showed here today, and thank you for your attention.
Great. Thanks very much, Dr. Young, for that presentation. I'm hoping that we'll be able to see Dr. Young. Oh, great. I can look down there too. Thanks for your presentation, Cora.
Thank you. Hopefully you can hear me okay.
Yeah, we can hear you. So I was curious, one question to start off with is, how do you think climate change might affect the processes that you're talking about with changing temperatures, melting ice? How might that tie into the cycles you've talked about?
Yeah, so that's a great question. One thing that we know will be an impact of climate change is the release of pollutants that have accumulated in ice. So I think that will be very relevant for these chemicals. We know that they're accumulating in ice, so we would expect that their release could have an impact on environmental levels in remote regions in particular. In terms of, there's lots of potential impacts of climate on ocean currents as well, which is beyond my expertise. And certainly there's impacts of climate on the oxidants that are necessary to actually degrade these chemicals in the atmosphere as well. So I think that's something that we certainly need to explore further.
Okay, great. Thanks for that. I think we'll start with any questions here in the audience in Providence.
I'll ask one. Thanks. My question was, do you know anything about the potential transformation pathway of these precursors into these really long chain, I think you said C14 and greater compounds? Very curious, thanks.
Yeah, so the transformation pathways are chain length independent. So when we find longer chain acids, that just means we started with a longer chain precursor. So for, I know residuals in commercial products up to, I think, 22 fluorinated carbons have been measured. So what we would expect then is these longer chain acids are derived from some of these longer precursors. So for example, if we started with, like, let's say the 22 FTO, so 20 fluorinated carbons, we would expect that we could form carboxylic acids that had 21 and 20 carbons. Those would be dominant products. So we haven't found those yet, but that may be detection limit. That may be because of detection limits, or maybe because of the abundance of those precursors. But yeah, the mechanisms are chain length independent. So the same mechanisms that lead to the atmospheric formation of PFOA and PFNA are the same ones that could lead to the atmospheric formation of these longer chain PFCs as well.
Okay, great. Thanks for that. Reiner, I see you have a question here in Huva. Did you want to ask that with the microphone here?
I guess it's thanks, Cora. Fantastic talk. 6 years ago, I had to give your talk at FLUOROS in Colorado. I think you did a much better job. So I'm glad we had you. My question was a follow-up to Matt's. Basically, is there any detection of these longer chain FTOHs or whatever else there might be precursors? Actually, have you found them, or is it just hypothetical because we see the degradation products in biota?
Yeah, that's a great question. So the longest FTO that I know of that's been found in the Arctic is the 10-2. There are, of course, detection limit problems with that. And of course, for the longer chain, because they're more bioaccumulative, we would expect that the atmospheric levels wouldn't have to be as high to sustain the levels that we observe in biota. And just kind of a sidebar to that is that doing these measurements in the Arctic and other remote regions is very challenging. Our methods right now, almost all of them rely on high volume samplers. And those, of course, require power. And it's hard to find a site that's truly remote that also has power. So even Alert, where a lot of these excellent measurements have been made, this is a Canadian Forces base, right? So it's not necessarily pristine in terms of PFAS. So to my knowledge, they haven't been measured. But I think this might be more of an analytical challenge than necessarily that they're not there.
Okay, we've got another question here in the audience.
Hi, thanks for your talk. I was wondering if, when you were talking about linear and branched isomers, if environmental fractionation has any impact and how you would assess that.
Yeah, definitely. Great question. So we would expect fractionation in environmental processes to affect the isomer ratios that are observed. Now, to my understanding, this isn't my area of research, but to my understanding, the fractionation there is fractionation, but it's relatively, it's on the few percent level in terms of the effect on the differences between the isomers is on the order of a few percent. So if we're looking at, something that's purely linear or just a few percent branched, it's likely that these environmental fractionation processes couldn't account for everything. But certainly this is a concern, and this is one of the reasons that using multiple different environmental forensic techniques is useful when we're thinking about these transport pathways, because each individual technique does have its own caveats, right? So combining them all together presents a much stronger picture of how we expect of the transport pathways for these chemicals.
Thanks for that. Looks like we've got another question in the back.
Hey, great talk. Thank you so much. So I have two questions. One of my question relates to, in your slide showing the oxidation pathways, you had mentioned that there's equal proportions of N and N-1. Are any of the fragments produced through like the N-2 and N-3, are ratios of those diagnostic at all? Of the, those other compounds that are formed? And then my second question is, what is on the horizon for you in terms of other things that are being transported to the Arctic? I saw a question from Mark as well of, are you seeing any vinyl ethers? Like what else are you keeping an eye on in terms of neutral transport? Thanks.
Yeah, there's a lot in there. Okay, so I think the first part of the question, now I've forgotten the first part. The second part of the question about new chemicals is a really good one. To date, I haven't been doing any Arctic monitoring of the air myself, but the deposition samples that we have from the Arctic that include ice cores, we have been looking for novel PFAS in them, and so far we haven't found a lot, but we are looking there to see if there's any evidence for transport of these chemicals. To my understanding, there isn't too much known about the atmospheric degradation of some of the new neutral chemicals, so that's certainly an area that could be explored. And I've learned a lot from this conference, actually, so that's something that I would be interested in following up on. And I can't remember the first part of the question. I'm sorry, maybe Laurel can help me out.
Did you hear the question, Cora?
No, I didn't. Sorry.
Oh. One more time, again.
Take three. Cora, can you hear me now?
I can hear you now.
Great, thank you so much. So the question just related to, on your slide as you're showing mechanisms of transformation
right.
of some of those carboxylic acid precursors. You showed that they're primarily produced in N and N-1, and then the N-2 and N-3 are kind of lesser components, or however many are in that series. Are the ratios of those lesser transformation products at all diagnostic as well?
Yeah, so we haven't found them to be so, partly because the higher yields of those N and N-1 make it a lot easier to use them as diagnostic tools. And once we get down to the shorter chain PFCAs, there are so many different sources for them that it becomes very muddy. And especially for the really short ones, which we, like TFA and even the C4, the perfluorobutanoic acid, we expect them to be formed from basically all fluorotelomere products, and there's CFC replacement sources as well, so it becomes very complex. So we haven't used that as a technique.
Okay, Emily?
Hi, can you hear me?
Yes.
Okay. So going back to your portion about Gen X, I mean, we can reasonably assume that's coming from one particular manufacturer. Were you able to figure out the location of the manufacturing based on deposition?
Yeah, great question. So those types of, sort of single production location chemicals could be very useful as diagnostic tools. So far, we haven't seen any evidence of deposition from the atmosphere to the Arctic, where my group has been looking, of Gen X. And we're going to keep looking, of course, but it looks like contamination so far has been mainly through direct sources. Which I guess is good news. I don't know.
Another question that kind of builds on that. In consideration of global efforts to control and regulate PFAS, the environmental forensics methods that you've talked about seemed helpful, and maybe this is when we've got more than just one source. Could you please comment on how that would help enforcing the polluter pays approach? That is, countries or communities could benefit from the science to identify where pollution is coming from.
Yeah, that is a really important question, I think. And it is a bit tricky because the way to effectively do that is we have to be able to know where the pollution comes from. So as we just heard from the last question, something like Gen X, that might be relatively straightforward, but for something like PFAS, that will be quite complicated to know where that pollution originated. So we have seen, one of the things we can do, and I didn't mention this in my talk, is we can use temporal trends. So from things like ice cores to try and figure out when, that pollution happened or where it came from, and whether regulation has an effect. That's something else we can do with temporal trends, but that is tricky, and that certainly would require some thought. That's a really great question. I'm going to think more about that.
Okay, great, thanks. There's another interesting question that came in from Matt Dunn. The heavier a neutral compound is, the more likely it is to fall out of the atmosphere early. Is there a chance that these are falling out over coasts or oceans, degrading to C14, and that is being taken up by ocean transport up there? Would that explain the lack of air detection?
Yeah, so that's a good question. And I mean, of course, if things are transported through the atmosphere and then deposited, they can always be deposited to the ocean. That's certainly true. And also, one thing that a question came up in one of the earlier sessions about migration of animals. So, does the fact that these migratory seabirds have long chain PFAS, did that come from their exposure in the Arctic or when they were, another part of their migratory pathway? That's not really clear. I think there are many ways that some of this data can be interpreted. Yeah.
Okay, great. Well, I know we have a few other questions, so I hope you'll stick around for the panel discussion, and we'll have more for you already in line. So thanks again, Cora.
Thank you.
All right, we'll move on to our second presentation now. Our second presenter will be Dr. John Odland. He's a professor of global health at the Norwegian University of Science and Technology in northern Norway. He is a specialist in OBGYN and epidemiology, and for 30 years he's been working on environmental and reproductive health in the Arctic. So now we'll see John's presentation.
My dear colleagues, welcome to a small introduction about the Arctic situation on health and environment. I'm a doctor, an epidemiologist, obstetrician. I've been working in the Arctic for a few decades, and at the moment I'm a professor of global health at the Norwegian University of Science and Technology. I have many years in my career at the Arctic University of Tromsø and worked a lot in the Arctic areas, especially in the Russian areas. And today I will talk a little about the levels and possible health effects and what we can do. These two pictures, they show very much some of the emerging problems in the Arctic. The polar bear to the left has moved into a house in a deserted village in the north of Siberia at the coastline of Russia, the Arctic Russia. And in these areas, there are many environmental refugees because the villages are actually sinking. So when they cannot live there anymore, they have to go away. The animals, polar bears, take over. So this nice little guy is living in a deserted house in the coastline. On the other side, we have what we call the Arctic dilemma. We have contaminants coming into the Arctic, accumulating, but they accumulate in the dietary chain. This babushka is giving healthy nutrition to the baby. The problem is that she is also giving the baby contaminants, because the contaminants, they often follow the lipid-soluble part of the diet. And this is an emergent problem that we need to take care of in the Arctic and also in the global dimension. So these are just very good symbols of what we are dealing with in the Arctic. We have a good program working on assessment of all kinds of effects on environment in the Arctic. The Arctic Monitoring and Assessment Program is a subgroup of the Arctic Council. That is a political organization with all Arctic countries, and also now including other countries like South Korea, Singapore, and Japan. And even the European Union wants to come in there because the Arctic has an increasing importance in many ways in the global perspective. The Arctic Monitoring and Assessment Program, where I've been chairing this health group for a few years, and we have monitoring a lot of different samples from air, water, snow, ice, plankton, invertebrates and fish, mammals and birds, all the way up to the humans, the dietary chain. So we look at different organic pollutants, inorganic pollutants, also essential elements, climate change, and what the effects of the oil and gas industry. So there are many quite controversial questions in the political situation here. So our work is not political. It is to give hard facts and evidence to the decision makers, stakeholders, politicians, and also to the whole background. And our aim is to have improved the health for the humans, the Arctic. And to have a good health for the humans, we need a good health in the environment. It's quite logic and very simple. The geographical coverage is not related to the Arctic Circle, because the climate is making the circle very different in the different parts of the Arctic. Like in Norway, we don't have any Arctic towns at all. We might call Vardø in the far northeast as an Arctic based on the temperature, but the rest is quite moderate because of the Gulf Stream. But in Siberia and in northern Alaska and northern Canada, we have the temperature zone that is going far beyond the Arctic Circle. So these are things we need to take into account when we assess the situation in the Arctic areas. Even so, the environment and the moving of the contaminants, it's not the geographical issue, it's a global issue. We simply cannot look at the geographical borders because the Arctic plays a fundamental role in the circulation of water in the oceans of the world. Every winter, several million cubic kilometers of water sink to deeper layers, which move water slowly south along the bottom, back again, and so it's an endless circulation between cold water, warm water, and the shift is also making a big movement for all kinds of the natural things like the contaminants following the streams. So that means we need to know the situation in the south, in the north, in the east, and in the west. That means we have done quite a lot of work now to compare. We are making monitoring programs, and if you have compatible data with good laboratory service, you designed studies all the way from the sampling to the background information up to the human levels. So these are levels in the blood of the PCB family, where the PCB 153 is actually 70 to 80% of the total PCB exposure. And you can see that the Greenland and the Faroe Island are very much exposed to this. And that is because of this, the streams of the air, water, and so on. They go to the north, follow the fluctuation to the northeast. So it means that in the southern hemisphere, the situation for these global pollutants is very different. We have done studies all the way south in Argentina, showing that the situation is totally different, because it's only more local contaminants we have in the southern hemisphere, because the streams are much weaker going to the south compared to the north. That means it's very different for the DDT, DDE family, because you can see in Mexico, the Caribbean, also in South Africa and South America, it's much higher concentrations, even if you can see that in the north of Greenland, we have also some global exposure. But the exposure in the southern hemisphere, that is mostly a local pollution based on a lot of use of pesticides and local sources going into the diet, going into the body, that you can read out of human blood. If you look at some other like HCB, HCH, these are also pesticides, and you can see some strange patterns, like in China, very suspicious of what we can call moonshining. That they make local remedies for pesticides and for protection of vegetables and so on. So we can read out quite a lot interesting out of blood samples around the world here. So, but also partly it's a global contamination to the north, as you can see in Greenland, but there is also local contamination in the Mediterranean. So these global overviews can give quite a few interesting perspective of what we are dealing with. But in the north, we have another problem. We have industrial waste sites in the vicinity of indigenous Arctic communities. At the Arctic coastline, especially in Russia, where I have been doing my studies, but also in the north of Norway, Canada, Alaska, and so nobody is innocent here. It's the industrial world that is imposing pollution on the indigenous people. And of course, that's one problem if they just leave a few 100,000 of PCB bottles. But this is what happens when we leave these barrels there. They are eroding. The contaminants move into the ground. In the ground, the indigenous people have store their food. Some old people in the indigenous communities, they have sky high the levels of PCBs. But is that the problem? Maybe not so big problem, because people who are old, they don't want children. But of course, we talk about hormonal mimics, and we talk about the cancerogenic immune system imposed and so on. So we have to be very careful where we address the public health advice. And of course, that is for the vulnerable population, for the young people, for the pregnant women, and those people who really need to be prevented from some hormonal disruption and endocrine impact. So that is the background for the Arctic dilemma. The Arctic dilemma is that we have the traditional food that is the most healthy for a cold climate. It's also that with the highest content of contaminants. like the PCBs, like the DDTs, also even the fluorinated compounds, as we will talk about later. So that's why we need to have a good dietary advice here. The worst thing we can do is just to move to what we call junk food, just for imported food. That has happened many places in the Arctic as well. Changing the health problems into something what we call a modernized health with increasing diabetes, increasing cardiovascular disease, increasing cancer. So these are problems we really need to take seriously. So this Arctic dilemma is very symbolic for the situation in the north. And of course, if you go all the way back to the conception, from this conception, this is a fantastic picture of the Swedish photographer Lennart Nilsson, showing this the capsule at this moment is fertilized, the capsule is closing, tail is lost of the sperm. Behind this poor sperm, there are 50, 60 million sperms dying in the next few minutes. So it's always a question to the fertilization is a quite a brutal venture. But then, of course, a lot of things can go wrong. And then if we go deeper into that, we should not do that today. But there is a reason why maybe 15 to 20% of the started pregnancy end up with a spontaneous abortion. So that is why it's so complicated what happens from this time of fertilization, the first couple of weeks. And that's also why we have so much focus on the first part of the pregnancy. That's where the fetus is most vulnerable to external exposure, including contaminants, including antibiotics, including different external exposures. So that's why we want to eliminate and do what we can to reduce the contaminant burden of populations. As if you start on the top here, these are from the AMOP recent report that I will come back to. From the early 90s until just a few years ago now, we have recent evidence that the DDT, DDE is going very nicely down. The PCB family and the oxychloroquine are also nicely going down. So at the same will show if the pregnant women in Canada, we see this nicely reduction. The problem is, of course, that these compounds, they have very long half-life, so we'll have to deal with them in nature a few generations ahead. But then we have the problem with the fluorinated compounds, and that's also the focus for this conference. And we can see that the recent levels we have all the way in the Arctic, we have taken out the Swedish studies as an example. We can see that these different fluorinated compounds, they are increasing in concentration. They are not going down. And that could have serious health consequences if you are not really aware of that. So what I present to you today, that is plain monitoring, just to show what is the real situation for people in the Arctic. And that's we are doing very well with the DDT family, the PCBs, and so on. But we have a problem with the fluorinated. And as you can see also here, this is from Alaska, Yukon, Nunavik, Faroe Island, Norway. These are adult population, and they also have the pregnant population of the Nunavik, Canada, and the Alaska concentrations that are very high on this PFAS. PFAS, that is really the big issue here. And you can see that the pregnant in Greenland, they have very high concentrations. And then we can talk about possible health effects. And that's where we want to have a discussion. That's where we want to point out the way forward. So now I just want to switch into some recommendations from the recent AMOP health report, Human Health in the Arctic 2021. It's so fresh that I just have to cut out some of the basic messages from the report that is not even published yet. So we have recommendations, of course, to conduct more studies to reduce uncertainties in the estimates of health risk. And we really need to develop new risk assessment methods and models. And also study protocols to may have a really compatible studies all over, because as I mentioned, this is not something we can do in one country. It's a global issue. And also we cannot only do in the Arctic. We need to have a collaboration with a global perspective here. Actions to reduce health risk, the evaluation, assessment all the time, because there is not one compound that is living alone in nature. It's a mixture of different compounds. Even the metabolites can have different biological properties. Like if you look at DDT and DDE, there's an example of that. And we must do a lot on communication and risk perception to how can we do this in a public health perspective. It's so important. So the understanding of health impacts without destroying the local culture, local dietary habits, and so. So we need also multidisciplinary research on, we put on critical questions on what we call on a different environmental problems. Like we cannot only look at humans without looking at air, water, animals, birds, fish, and so on. So this is really what we gone into the One Health concept. It has emerged as a useful concept to approach and address multiple challenges related to environmental challenge. Human, environmental, and animal health are interdependent. And to bring this together, we need new collaborative studies to investigate levels of chemicals, routes of exposure, health effects, and so on. These are so important things to bring up for future research now. So what the report says to the politicians, because our role is to bring up the hard evidence, but we need to bring it to the decision makers, to the keys, to the stakeholders, politicians, and so on. We need to follow up the Stockholm Convention, the Minamata Convention for POPs and mercury, and the Arctic states should take steps to reduce or eliminate chemicals. But it's a very challenging situation, because every year we also find new ones coming up. And those we still do not know the real long-term effect on human health. Even if it looks like it's tested nicely in the laboratory, what do we know about the long-term chronic exposure? We have a lot of things we don't know yet. Beside that, we have to take care of the people who are living now to get the best out of traditional country and local and store-bought Western diets, to look at what is the healthy choice. If we cannot think about contaminants without thinking about nutrition. And of course, we see now in the new generation tendencies to having low vitamin D levels. Even in Norway, in our cohort ongoing in the north of Norway, we can see that there are very low iodine levels. And of course, iodine is so important for the thyroid, for the metabolism. And if you have too low iodine levels in pregnancy, they can have a serious impact on the fetal development. So we have new challenges coming up when we're trying to reduce or eliminate the contaminant situation. So food insecurity in Arctic communities is the main issue for us. And also we need to expand efforts to collect data on exposure, dietary transitions, and health impacts. The dietary transition is a serious problem now. Maybe we were so uncritical to that the first time 30 years ago, when we started to know the high levels of contaminants like PCBs. But there is an effect of all changes, because the diet is based on hundreds of years. Suddenly, you're giving different dietary advice. We cannot do that. We need to have a holistic approach to everything we do concerning the human health. And one of the things we are so concerned about now is this increase of the fluorinated levels in the Arctic. I will now just finish with showing some perspectives of my work in the Russian side of the Arctic Circle. If you talk about the Arctic Circle, we very much forget that we on the western side, we are only 50% of the Arctic Circle. And if you have 50% on the Russian side, and we can say that permafrost is covering almost 70% of the Russian territory in the north. Of course, then it has serious consequences if the tundra is melting. Back to my polar bear, who is living where the people had to run away. Siberian tundra is now warming, is causing frozen permafrost to melt. It's softening, subsiding, forming watery thermokarst. That is the way to look at how will the polar bear survive here for the food availability. How will the fish survive with different, the rivers are changing, water is changing. What happened to, if you increase the temperature, what happens to the vectors coming up there? The insects, they are very much depending on temperature. And that, of course, they bring new viruses, new bacteria, and so on. So this is really something we need to observe, we need to handle, and we need to have a proper assessment. And that is why I'm so concerned about having good monitoring of nature and good biomonitoring for humans. And that's how we can detect like the fluorinated compounds increase. So this now I leave it open for a discussion. Tusen tack.
Okay, great. Thanks very much for that presentation. Do we have Dr. Odland online?
Yes, I'm here. It looks like
Terrific.
I'm here.
Thanks very much for your work and for your presentation. I was going to start off with a couple of questions. I noticed that on one of your graphs you showed that pregnant women in Greenland had higher levels of PFAS than women in other regions. Do you have a sense of what the major reasons are for that? Does that have to do with reliance on local foods or kind of the environmental fate and transport and accumulation?
Due to the data, we know that this is really a global issue. It's not so much about the diet, but also about what is coming in, especially through the ocean to ocean transfers. So it's partly to come into the diet, it has to do with if it's water soluble or lipid soluble. The fluorinated are maybe in a special situation now, but mostly it's coming in through the what they call marine mammals. Those also bring it into the diet. So maybe that's the good explanation without having absolute evidence at the moment. The point is that this is what I showed you today. That is really monitoring what we are doing now, and we need to know a little more about the sources. But it looks mostly like it's coming in through marine diet, as we know, per date. Thank you.
Okay, thanks for that. I also wanted to ask, you'd mentioned about local industries as being sources of pollution. How much is known about sources of PFAS, local sources of PFAS contamination relative to the sort of global transport that we've been also talking about?
The local production in the Arctic is very low. So I think this is what we show in the blood samples now from the Arctic. That is really a global issue coming from the industry and in the central areas of Europe, from the US, from Canada, and it's really it's the north flow. There is a principle here also for the fluorinated compounds. Like we see, I think we have much lower concentrations in the southern hemisphere that is going into the human body. So this is really something that we can address to the industrialized parts of the world. And our worry now is that the Arctic is slowly getting more industrialized. And of course, they find enormous resources now of minerals. More the oil and gas activity is increasing, and there are coming in new industrial actors from other countries. Those want to join this Arctic Council. So we really need to address this before getting into the huge problem. And even the way when we talk about green energy and then new sources of energy, you need minerals for that. And where do we find the minerals to make batteries and so on? We really need to find it somewhere. And the place they are really exploring now, that is in the Arctic. And there is a heavy political pressure now to get into the Arctic to utilize the resources that find also for the green energy. This is really a contradictory part of the new consequences of climate change. Thank you.
Okay, thanks for that. It looks like we've got a question from the audience here.
That sparked my interest. This was a fascinating talk, and I'm wondering, in particular, if you have any contaminants that are unique to the oil and gas industry or to mining that you're seeing in some of these populations up in the Arctic.
Yes, you can mention quite a lot of them, and what I'm concerned, of course, about the fluorinated, but I'm also very much concerned about some of the toxic metals. Also, the heavy metals like mercury will be an increasing problem in the time to come. But another thing I had no time to mention now was the emerging compounds. So we are in a way working where the industry is one thing and our research is one thing. It's like being a detective and find out what they really use for things. And one example I can mention is that in the north of Norway, now we have a study where we did pooled samples of blood to look for so far undetected the compounds. And we found some, we knew that they were there, but we were surprised that some, for instance, like medium chain chlorinated paraffins, they're really increasing the last 15, 20 years. So this is a good argument. We really need to do a good monitoring here, also emerging contaminants. That some of the heavy metals, like what you need for batteries, and what you can get out of the side project of oil and gas. This is really, I would call it maybe fascinating, but also a little scary that there's a lot of effects here that we don't really know yet. But these are examples of what we are dealing with now. Thank you.
Okay. Any other questions from the audience here?
Very much for your excellent presentation. You introduced the Arctic dilemma about having the reliance on traditional food that are heavily contaminated, but they're also best suited for living in the Arctic. Yeah. And you spoke about the dietary transition. But so what do you actually recommend? Because most communities can't grow greens year-round. They don't really have access to less contaminated cattle or whatever you might have. So what do you actually recommend, given that ideally they need proteinaceous, lipid-rich diet, and yet it's so contaminated? Is it a matter of restricting it to more ceremonial times and relying on some other diet in between, or what do you suggest?
I think I can use Faroe Island as a very good example of what we want. Yeah, and we can use also Greenland as a good example of what we don't want, because when we started to discover this hormonal mimics in the diet 30 years ago, they started to skip the traditional diet that was very based on fatty food, and they started to import imported food, a lot of carbohydrates and so on from Denmark, leading to we lost a lot of the traditional food, and ended up with a new increase in diabetes and cardiovascular disease. Consequence of all these things is that now we can stratify the dietary advice, that we need to know as much as possible about the content of contaminants. Like in a Faroe Island now, they have developed a strategy to reach the young people, the pregnant women, and also to give different dietary advice to older people, because there are certain whales and marine food that is high in contaminants. Don't eat that while you're young or pregnant, but you might eat it if you are old and don't need to think so much about the contaminants. And so, and some of these marine mammals and marine food is low in contaminant. It depends on how much fat there is in the body and so on. And so we can really give detailed dietary advice without skipping the historical good diet. So that has been, we have done a lot of this the last 30 years, and things are really improving now. So we can see that, that for the young people now, the levels of contaminants in humans, that is decreasing now. So that is a very good development without skipping the historical diet.
Thanks, John. I have a follow-up because I was just on the Faroe Islands, and I assume it's maybe similar to some places in Norway where you can go to the supermarket and get anything and everything at any time of the day or season, but I don't think that holds true for some other indigenous communities that are much more cut off from regular supply chains. So what choices do they actually have then?
Well, still we can do something. I agree that if you go out of the Faroe Island, out of Norway, you get into the indigenous populations in the north of Canada or north of Russia. They don't have the choices. But we can help a lot by analyzing the food items and also give advice without only move to imported food, because that is so much worse for the health, especially also for the young people or for the pregnant. But I agree there are still parts of the Arctic where we simply cannot do the optimal food choice still. But I think we are working on it in the public health perspective, all places, even in the smaller parts, populations of the Arctic.
Okay, great. Thanks for that. I had another question. I know you've been studying persistent organic pollutants in the Arctic for many years and have followed cycles through of pesticides and PCBs, and now we're talking about PFAS. Do you think that there's something different about PFAS, or is it just sort of the next chapter of this story, and thinking ahead, how can we avoid this happening with some new type of contaminants in the future?
The only way to avoid and reduce is that to take care of the sources, and those sources, they come from the south, from the developed parts, like the industrialized parts, and how they go into the body. It also always depends on if there is a lipid solubility or a water solubility, and that means so the different food items, like the PCBs and the fluorinated, they go into different food items based on the solubility. So we really need to monitor also the content of the different food items. But in general, the fluorinated, that is really the next chapter of the same problem, to my view. It's not nothing new. It's just that the new compounds, they differ from the old, but still the same problem. And what will happen after the fluorinated? I think there will be a lot of the new emerging contaminants we don't even know yet. The new perspective is that most of this new, they might be controlled, they might have a shorter half-life. We have to use different monitoring, like maybe urine or hair or whatever we can use to monitor, but it's really a part of the same, it's just a new chapter of the same story.
Okay, thanks for that. And it looks like we've got one more question for you on HUVA here. This says, I'm actually surprised that some of the developing countries that you showed had considerably lower contributions of PCBs, et cetera, specifically because their environmental regulations are commonly poorly implemented. My question is, could the contribution of POPs be more related to industrialization of a country rather than environmental regulation efforts?
It all depends on what type of compound we are talking about. Like these global substances, they follow the big air and water fluctuation, and then of course they go to the north. So even developed countries in the south or developing countries in the south, I have done studies in the southern Africa, and even if they are very poor countries, there is no control. This global contamination is very, very low. But instead we find this pesticide problem that is so much higher in these areas. So it all depends on what type of compound we are talking about. And there is an increasing awareness now in the southern Africa also about pollution. And we are doing studies now that needs to be done, because what happens when you develop a population where we have an increasing middle class? Yes, they all buy a car, and I have never seen so bad traffic as I've seen in some of the bigger cities in southern Africa. So the pollution is coming out there because of the lack of control, as I totally agree on that. But that is the local pollution. The global pollution, that is really going to the north. Thank you.
Okay, thank you for that. And I think we've got one last question that you can weigh in on here. Has there been any correlation discovered between PFAS levels and distance between Arctic communities and firefighting activities or landfills or other sources of PFAS?
Yes, those the skiers, with this, what they use on the skis, that's one thing. The firefighters is one thing. And then, of course, but they are what we call cases. When we talk about the Arctic global pollution, that is still lower. But I think the highest levels we find on really exposed groups of people, like the firefighters. I don't remember exact how many micromole per liter, but at the moment, but they are cases. They're really higher exposed cases. Those are higher than the general population of the Arctic. But still, we are working on how this will have impact on the hormonal mimics. And of course, firefighters who are maybe adult people who are not pregnant, they might have a better situation also even if they are higher exposed. We are working with the young people, with the pregnant women, and those who really can be really vulnerable. And then, of course, we are very concerned about like a chronic exposure, even if it's lower than those extreme cases. Thank you.
Okay, well, thank you again for your presentation. I think we'll give you a round of applause and then move on to our next presentation. And I hope you'll be able to stay for our Q&A at the end panel. Okay, so we're moving on to our third presentation, and we will be hearing from Vi Waihi. Born in Savunga on St. Lawrence Island in Alaska, Vi is Environmental Health and Justice Program Director at the Alaska Community Action on Toxics and a Yupik grandmother. She serves as an advisor to the National Institute of Health and has been awarded for her work by the Alaska Native Tribal Health Consortium. She is one of 26 members chosen by President Biden and Vice President Harris for the new White House Environmental Justice Advisory Council. So it's an honor to have Vi with us today.
Hello, my name is Bangunak. I'm the daughter of the late John and Della Wagai. My grandfather is Wagai. He is from Punguk and Gangerame clan. My grandmother, Ayoklik, is from the Uwalit clan. I am from Sivokak, a traditional name for St. Lawrence Island. I'm a Sivokak Yupik grandmother. I live and work here in Anchorage. on the unceded territories of the Dena Aina peoples. I am from Savunga on Sivokak, which is located on St. Lawrence Island. The title of my talk today is Contaminated Without Our Consent, PFAS Threatening Food Security and Health of Our Yupik, Sivokak Yupik people. Community-based research on Sivokak, protecting the health of future generations. I am a Native Villages Sivunga tribal citizen and Environmental Health and Justice Program Director with Alaska Community Action on Toxics here in Anchorage, Alaska. A little bit about ACAT, the organization I've been with going on 19 years this year. We believe everyone has a right to clean air, clean water, and toxic-free food. Our methods include community-based and participatory scientific research, public education and training, community organizing and advocacy work, but ultimately to achieve policy change at the local to international level. And we do support adoption of the precautionary principle, protective of human health, better to be safe than sorry. This is the image of Annie Aloha. She was a community health aide on our island for 25 years. Annie lived and worked at Northeast Cape, one of the communities where a formerly used defense site was established during the Cold War. St. Lawrence Island was an important strategic location for the military that came during the late 1940s and left in the early to mid 70s. Annie started noticing a lot of cancers in our people. She was a very keen and trained observer about the health of our people. She started noticing high rates of cancer among our families that lived, worked, and camped at Northeast Cape. Not only these cancers, but also low birth weight babies and miscarriages, especially our families that, as I mentioned, lived, worked at Northeast Cape. We remember her knowledge and forewarning as a respected community health aide and elder. She inspires our work today. We now know that the Arctic has become a hemispheric sink for persistent organic pollutants. Indigenous people in the Arctic are more exposed disproportionately to toxic chemicals, and children are at higher risk. And those of us Arctic Indigenous people, we are some of the most highly contaminated population on the planet because of our reliance on our nakapik, our traditional foods. Because of these persistent organic pollutants that contaminate and build up in the blood and breast milk that are among some of the highest of any population on the planet. These, the global transport of POPs come into the Arctic and now are being exacerbated by climate change. And we now know that the Arctic is warming 3 times faster than the rest of the planet. Melting Arctic ice, glaciers, and permafrost are releasing these POPs that have been sequestered for decades. This is another route of exposure to our environment, our lands and water, and our people. And microplastics are into our environment and also into our food web. We now know that the Arctic has the most microplastics of any oceans in the rest of the planet. Sivokak is located in the northern Bering Sea. It is about 100 miles long, 30 miles wide. As you can see, it's in northwestern Alaska. Our creation story is that the creator put his sand into the ocean and pulled up some soil and dirt from out of the ocean and wrung it out. And as you can see, it seemed like it's been wrung out, Sivokak. And we have two communities on our island. Gamble, which is in the north western, is older of our two communities. We have about 800 people there. And where I was born and raised is Savunga, in the middle of the top, as you can see. We have about 900 people there. And on the very bottom right is Northeast Cape, one of the two formerly used defense sites on our island. At one time, there used to be 10,000 of us in 32 communities, but due to introduction of Western culture, generational trauma, decolonization, our numbers have greatly been declining. However, we are still here. We were very fierce warriors. Mainland tribes feared us, and we have inhabited our island since 500 BC. These are just some of the images of our way of life. My people have lived as one with our lands and waters, with utmost respect as one, taking only what we need from our lands and waters. A way of life my people have lived for many generations. As you can see on the top left, there's a young man collecting myrrh eggs. These young men scale these cliffs to collect bird eggs. And the woman on the bottom right is sewing a traditional skin boat. You can see the image of it on top. Historically, we use skin boats to go hunting in the northern Bering Sea. And on the bottom right, you can see images of our young men singing and dancing, or atok. These songs and dances have been passed on for many generations. We are intricately tied to our lands and waters and the wildlife that have sustained our people for many years. And these are some images, historical images of my immediate family. You see the images of our grandmothers on the bottom right. The one you can see on the bottom row, second from the left, is my mother's mother, Aka, the one with the most white hair. And the images above our grandmothers of our young men and women continuing to sing and dance between the arches of the bullhead whale. The two young men are singing and drumming while the women are dancing. Many of our dances tell of our way of life. And the image to the right of these images of my immediate family, my Apawakayi and my Ningyok grandmother, Ayuklik, my parents and mother. This is a very old picture. It was even before I was born. My father, Kayak, my mother, Singik Parmi, and my older siblings. I was the youngest of 6 siblings. My grandparents had 12 children, and only 4 survived. My grandfather was an orphan, and because of a great starvation and epidemics, they decimated the numbers of our people. But he survived. He was a survivor. And my grandmother was a midwife who delivered all the babies in our community. When I have struggles within my job or in my life, I think of my grandmother. I imagine her strength she had as she's delivering all the babies in my community. She is burying her children. So we come from a long line of strong people, and I now believe I am where I am supposed to be, that this is my role, to be the advocate and voice not only for our ancestors, but my people and our future generations. As I mentioned earlier, our Indigenous people knowledge is at the foundation of our community-based research. And these are just some of the quotes that I'd like to mention. This is from Merle Apasangak from Native Village of Gamble. His quote goes, We have always been a vigilant people. Our community-based research enables us to be vigilant at the molecular level. And the one under that is from Delbert Pangau. You see an image of him holding up a dead bird that has washed up. We are overwhelmed with concern about the health harms associated with climate change, the loss of sea ice and melting permafrost, and the mobilization of chemicals and plastics. These are all interconnected. We are running out of time. And the final quote is, we don't just eat one chemical, we eat the whole fish. This is from a former board member with Alaska Community Action on Toxics. She is from Native Village of Port Graham. These are images of the formerly used defense site at Northeast Cape. It was operational from 1957 to 1972. There were above ground structures and debris removed in 2003, and approximately 123 million has been spent on the site remediation. However, these sites have never been properly characterized and have been prematurely closed. My people welcomed the military when they came. We were an important strategic location for the military. We were the eyes and ears of the North during the Cold War. Our men and women helped rescue downed planes and were very patriotic, but now feel that the military have turned their back on us. And the image on the top right is of Northeast Cape. It was a pretty big installation, several hundred personnel. They basically abandoned when they pulled out after the Cold War, and they abandoned everything. When they came, they brought everything they ever needed for 15 years. As you can see on the bottom image, thousands and thousands, and estimated over 300,000 barrels that they abandoned, rather than spend money to remove everything they brought to our island. And the image on the top left is of White Alice sites. There were many of these sites built during the Cold War at the top of Alaska and all along the western coast. Before satellite, they built these structures for early warning systems. And these structures were approximately 13 stories or higher. And they needed a lot of power, so they used transformers. And the lubricants that they contained had PCBs. We now know that PCBs, even though they've been banned for decades, are very persistent. And we have come to learn that not only PCBs, pesticides, heavy metal solvents, there were also massive fuel spills. And the military was very negligent. It's been very difficult to hold them accountable. There was even an agreement signed in 1952 with our tribe and the military that specifically mentioned the vulnerable locations at Northeast Cape, the seal haulouts, and basically for the military to remove all the debris and trash and that they brought in. And our tribe wanted the military to turn our lands back in the same condition as when we turned them over. And this agreement has never been honored. We have had a community-based participatory research on our island in partnership with our leadership and communities in Savunga and Gamble. And as you can see, we have done extensive community-based research as the burden of proof has been put on us. And including hours and hours of footage of elder interviews. We've collected the blood of our people. In the beginning, our people wanted to get our blood tested first to validate what we'd known all along, that the military toxics that were abandoned and buried on our island. We attributed all the cancers and low birth weight babies and miscarriages that Annie started recognizing in her people. My people now have 4 to 10 times higher PCBs than the average American in the lower 48. And all these samplings that you have done I myself have done them all. We hire and train local people. Our people are involved from the beginning. They help design the research and what data to collect, including plants and berries and our nakapik, our traditional foods. We've also done extensive community health surveys because our tribe petitioned ATSDR to do a health assessment, and these health assessments are inconclusive by design. They do not represent small populations with people lower than 100,000. And basically, the cancer registry blames cigarette smoke for all our cancers. So we had to do our own community cancer registry. But we've also not only looked at military toxic, but newer classic chemicals, including in the household dust for PBDEs and PFAS. Some of the health that we're noticing we had never seen before until after the military left include cancers, thyroid disease, the cancers associated with the military toxics, but also with PFAS include testicular and kidney cancers that are most strongly linked. Thyroid disease, diabetes, heart disease, I mentioned the birth defects, but other reproductive health implications. These toxic chemicals are endocrine disrupting chemicals. It affects our ability to conceive. It affects our ability to carry our children. It affects our children's IQ. We are now seeing and hearing of concerns of lower test scores in my community. So these are burdens we didn't create, and we are seeing illnesses and health disparities never seen before in our people, including PFAS, which are a class of 100,000 chemicals of per and polyfluorinated toxic chemicals. These are images of Northeast Cape, and these toxic chemicals are multi-generational. The military displaced our tribe and community. We had about 130 people that lived at Northeast Cape. And the health disparities and cancer crisis we're seeing now most are our children that lived and played at Northeast Cape, the contaminated Silky River. It's not a matter of if we'll get cancer, but when. And our Northeast Cape families are now seeing the most health disparities among the other groups in our communities. Some key findings concerning the sentinel fish species. We have found exceptionally high levels of PFAS, especially PFAS and PFNA, in 9-spine stickleback at Troutman Lake, suggesting a local source. We use stickleback fish. We collect them because they're indicators for reproductive health. As I mentioned, 70% of the reproductive health of the stickleback is same like humans, so they're indicator species. And the PFAS and PBDEs are present in both the stickleback and the Alaska blackfish. And the patterns of exposure in the stickleback suggest that both global sources and local contamination are important sources of our exposures. PCBs are disrupting the endocrine systems of the fish, as I mentioned, and decreasing expression of genes important in repairing gene mutations. And concentration in the male stickleback indicate exposure to estrogenic contamination, certain PCB congeners. This study demonstrates that even after site remediation, contaminants from the Cold War formerly used defense site in remote regions of the Arctic continue to be a potential health threat to local residents. widespread health disruption in the fish is consistent with the health problems in the people on my island. Stickleback are an important sentinel species. And these are some of the key findings. PFAS and PFNA are detected in more than 98% in the blood of my people and in the blood serum. And PFOA, was detected in 92% of the samples. And most of the serum PFAS are comparable to levels in the US general population. However, PFNA and PFUNDA are more elevated. We found that certain PFAS disrupt thyroid hormones. And although PFAS concentrations are on the low end of the ranges reported in other studies of Arctic biota, this study found that different patterns of PFAS substances in tissue samples. And we believe that this is a first report of PFAS concentrations in bowhead whales with higher accumulation of PFNA and PFUNDA. However, short-chain PFAS substances are also present in bowhead and occasionally are most prominent. Other previous research on traditional foods on our island showed that marine mammals contain high body burdens of legacy POPs, as I mentioned, such as DDT, HCB, PCBs, and pesticides such as chlorinates. The current study suggests that recent and current use of POPs, such as these PBDEs and PFAS, are also present in the marine mammal tissues. Exposure to POPs through Arnabic and traditional foods and local hotspots of pollution, including PFAS, remain a serious health concern for our people, but also of the circumpolar Arctic, including the Yupik people on my island. These are drafts of the concentration in household dust and in the blood serum of our people. And some PFAS, such as PFNA, as I mentioned, PFUNA are actually are relatively common in the serum, but uncommon in the indoor environment. This suggests that our diet is our primary source of exposure. These are burdens we didn't create, as I mentioned, but they are ending at the blood of our people and household dust. Our homes are closed in more during the winter. Our homes are more ventilated, and these are very persistent. They don't break down, and with these chemicals should not even be in the blood of my people or anybody on the planet or in the household dust. And it's alarming to learn that my grandson, who I am raising, has higher body burdens of some of these toxic chemicals as adults. And this is very concerning to me. These are images of the some other data that we have found on our island, including, I'm sorry, hold on. These are images of the TSH, which is the thyroid stimulating hormone. Which is produced in the pituitary gland, and it stimulates the thyroid gland to make more thyroid hormones as needed. Our findings are statistically significant for PFOA and PFNA and association with TSH. So this is some of the data, and those of you who might be more interested in our publications, I will provide contact information at the end of my presentation. This is a report of the investigative report of PFAS in Alaska. The use of PFAS-based industrial firefighting foam on military bases and airports has contaminated the drinking water of communities throughout Alaska, and PFAS has been found at over 100 sites at nearly 30 locations in our state. And at least 10 Alaska communities have PFAS in their drinking waters at levels deemed unsafe by the US EPA. And it's unlikely that the number of communities with contaminated water will grow as more sampling is conducted throughout the state. PFAS are contaminated groundwater, surface water, fish, wild game, garden produce, and backyard chickens in Alaska. Several Alaska lakes are now closed to fishing as a result of this PFAS contamination. And we have worked on legislation to address PFAS issues in Alaska. We have a House Senate bill and a Senate bill that would require greater protections for these communities by preventing and addressing PFAS contamination, including setting enforceable drinking water standards for a number of PFAS, as well as requirements for polluters to pay for safe drinking water and blood tests for people affected by PFAS contamination. A little more about the CBPR that we have done on our island. We offer a week-long college-credited course to train community people, tribal leaders, as you can see, grandmothers, youth, to conduct our own environmental and health research. We have had 3 in Nome, which is on the mainland. That is a hub for my people on the island, but 1 in our own community. And these are just some of the images of some of the training, including how to dissect stickleback and other hands-on training. Also, we do extensive organizing with our youth, women, and elders to achieve health, but also our basic human rights that are being violated by these toxic chemicals, whether it's military, our human right to health, our human right to food and subsistence, human right to free and prior informed consent, but also environmental and reproductive health and justice. And these are just images of some of the samples of our training. Our staff and volunteers have had opportunities to testify and meet with the UN Rapporteur on the rights of Indigenous peoples. The image you see on the top right with James Anaya, we had this training here in Alaska in Chicxulub, where women were able to testify. He was the Special Rapporteur on the United Nations rights of Indigenous peoples. This is our images of our tribal community forums and Indigenous women's gatherings. As I mentioned, this is an opportunity to educate and offer training to women and girls. As I mentioned, our wombs are our children's first environment, and it affects our ability to conceive, our ability to carry our children. In our culture, we honor our elders and our children. So these are the classroom components of the training on how to testify, how to contact your legislators. But also on the bottom is image of Dr. Linda Birnbaum and her husband's David visit to our community. She was the only high official with the National Institute of Environmental Health Sciences under NIH to come to a rural community. I served as a National Advisory Environmental Health Science Council, and every meeting she gave updates. And when I heard her talk about her report that she had gone to Indian country, I invited her to St. Lawrence Island, not knowing that it would occur. But it did happen, and it was an honor to have her. And just a few more slides I'm almost done here. And the language of the Stockholm Convention recognizes special vulnerability of Arctic indigenous people and is aware of the health concerns and particular impacts upon women and children, and through them upon our future generations, and the conscious of the need for global action. We bring Arctic indigenous people to the Stockholm Convention. We are instrumental in helping to ban toxic chemicals globally. We go there to be the conscience of the convention, and it is determined to protect the human health and environment of people globally. And just one last quote. These chemicals present in our bodies are passed on to our indigenous children and harm their ability to learn our languages, songs, stories, and knowledge. Ikamsi kayo vikamsi. It is an honor to present at this FLUOROS Global 2021 conference. I am honored to share our community-based participatory research project and give a community perspective. And these are a list of some of the publications as far as the research that we have done. Ikamsi kayo vikamsi. Thank you very much.
Great. Thank you very much, Vi, for being here. Very early Alaska time. Thanks for your presentation. I wanted to start things off. You've done so much great work with researchers and doing your own research. I was wondering what are, as we're mostly scientists here, like what do we need to know from communities like yours about how we can be better partners on research, and what questions and needs do you have for us?
Thank you. My name is Pawanak. I'm the daughter of the late John and Delaware, and I am calling in from the unceded territories of the Dana-Aina people. I live and work on Sivokak, our traditional name for St. Lawrence Island. I have to introduce myself traditionally. And, what do we do in science is not enough. In my community, we have a cancer crisis. It's not a matter of if we'll get cancer, but when. And since we are some of the most highly contaminated population on the planet, these are very alarming, not only to myself as a community-based researcher, however, to our leadership, our parents and grandparents. I was my parents and siblings, we were a family of 8. Half of us have gotten cancer in my immediate family. And we need to find ways to address the health disparities that my people are seeing, not only from the PFAS, but the over 130,000 chemicals that are made and produced, and only a handful are ever tested for their safety before they're put in the products that we bring into our home. We're being exposed in our home, workplaces, and communities like mine through our nakapik, our most sacred that have sustained our people since time immemorial. So I'm asking those that are in the room, we need meaningful change. We need to strengthen chemical and environmental laws that are most protective of the most vulnerable, including Arctic indigenous people, our children, our future generations, to hold polluters accountable, manufacturers accountable, to address the health disparities. It's not a matter of if we'll get cancer, but when. We need adequate health care. We are being exposed not only from one chemical, but a toxic stew. And it is very alarming to learn that some of these toxic chemicals can affect our children's ability to learn As I mentioned, how can we pass on our creation story, our songs and dances, our languages, our culture and traditions to our children if they cannot learn? You know, our wombs are our child's first environment. I've had 3 miscarriages, and I have been blessed with 4 sons now. Some of these toxic chemicals affect our ability to conceive, ability to carry our children, our ability to have safe births. So we need to make existing chemical and environmental laws as strict as possible. And the few handful of these chemicals that are tested for their safety, that number should be just as many as these toxic chemicals. These manufacturers are driven by greed and profit and do not take our human health into account. I am making a plea to those that can make changes. Go back to your communities and countries and attend meetings to make sure that these broken and outdated laws are updated and very stringent for my health of my people, but my grandson, granddaughters. Thank you.
Thank you for that. Do we have any questions here in the audience?
Presentation. I just wanted to ask, how do the levels for your people for PFAS compare to NHANES data or some of the average levels that we see in the United States? Or I'm not sure if, I guess it's a question for any of the, for you or Laurel. Are they similar, different, higher?
I believe that, go ahead.
Oh, did you hear the question?
Did you hear the question, Vi?
Yes, I did.
Okay.
I believe that some of the PFAS, as I mentioned in my presentation, such as the PFNA and PFUNA are relatively common in serum, however uncommon in the indoor environment, and that this suggests that diet is a primary source of our exposure. So we found it in 98% of our people that we tested, our reproductive age men and women, 18 to 45. And I'm not certain of how they compare to NHANES. However, they should not even be in our blood serum. And, you know, nobody's blood serum, whether it's my community or in the community where you come from, or anybody in the room where you are. So these are, to me, it is so unethical that these toxic chemicals are produced and made and never tested for their safety. We're being exposed without our consent. To me, that is what is most alarming. I can get back with you regarding how they compare to NHANES, but it's just that, you know, manufacturers need to be held accountable. To me, that's more important, but I can get back with you certainly. And if you can send me your name and contact information, and we can get back with you.
Absolutely, and I don't mean to, and I certainly don't disagree with what you're saying at all. It was just more from a scientific human health standpoint to understand the differences in exposure and help us understand, that's all. Thank you.
But what I can say is that we're being exposed through our nakapik, our traditional food. 98% of our homes eat only nakapik, traditional foods, a way of life my people have lived for centuries. And we're being exposed without our consent through our traditional foods. And we're finding high levels of PCBs in our marine mammals that sustain our people during our long winters. These toxic chemicals, I know this is a PFAS conference, however, the PCBs in our marine mammals is killing my people. They should not be in our nakabik, our traditional foods. And these toxic chemicals that have been sequestered in glaciers, permafrost, and ice are being even released even quicker. So these are burdens we didn't create. So thank you for that great question.
Great. Thank you very much. We've got another question here.
Good morning, Vi. Good morning to Alaska. Thank you very much for joining the conference. Rainer Lohmann speaking. I wanted to follow up on your discussion of the traditional diet that is so contaminated by PCBs, mercury, and now PFAS. In the previous presentation by John Ogden, he was talking about the switch in diet. So what does it actually mean for you and your family and your community? Are you foregoing sometimes eating seal, or do you keep going because it's traditional and it is what has sustained you for generations and generations?
My people, it's important to know that my people still feel the benefits outweigh the risk of eating our nakabik. And we are in a remote island in the northern Bering Sea. In the northern Bering Sea, everything is flown in. And a gallon of milk costs $11. So only 12% of the people in my community have jobs. And the median income is about $26,000 a year. So this is a way of life my people have lived for many, many generations. The only option, the food that is at the native store, are highly processed foods, packaged foods, canned foods. Very seldom do we get fresh fruit. But my people still feel the benefits outweigh the risk. It's our identity, it's our culture, it's our way of life. We have some of the most super packed traditional plants and berries and nakabik because of our short summers. So it is important that yes, we provide information on ways people may be able to reduce their exposures. We know that PCBs build up since they're fat soluble, build up in the blubber and rendered oil of the marine mammals, the bowhead whale, walrus, and 4 species of seal my people live off of. And that the meat is not as high, and that the land mammals they fare much better. So these are things that we've learned and we share, and when we are asked, we don't tell people what to eat or what not to eat. We provide this data so people can make their own informed decisions.
Okay. Thanks for that. I don't see any other questions here in the audience. I was wondering, it sounds like you have 2 types of challenges. You have these global pollutants that are used and manufactured far away and get transported to your environment in the Arctic. And then there are also local sources of contamination, such as the military base that you mentioned. So in your advocacy, could you kind of speak to the challenges of addressing both these local and global sources?
We've exhausted all efforts to hold the military accountable. There was even an agreement the military signed in 1952, which I mentioned in my presentation. These sites have never been properly characterized. We identified over 32 sites of concern. We've identified PCBs, pesticides, heavy metals. There was massive fuel spills, and we are now hearing in the national news about a massive crude oil spill that happened somewhere here in the lower 48. we have had spills up to 220,000 gallons that we know of, 160,000 gallons at one time. And the fish have never returned. Sucre River at Northeast Cape used to have one of the most abundant fishing locations on our island. We have no more seal haulouts, no more tomcod fishing. And there is no funding for remediation and site characterization. It's going to take an act of Congress. And our tribe was never party to the record of decision. So there needs to be ways to hold the military accountable. And the persistent organic pollutants, even though like PCBs, we can't taste them, we can't smell them, they are very prevalent on our lands and waters. And it is so, as I mentioned, I'm appalled at how human health and public health are not taken into account when these toxic chemicals are made and produced. Within 5 to 7 days, here's an example. A farm worker in South America applies a pesticide, and through air and ocean currents, it ends up here in the Arctic through air and ocean currents. And this is global distillation. So there needs to be investigation. These polluters need to be held accountable. These human health and public health need to be at the forefront instead of profits. And these are some of the struggles and concerns I'm bringing to the White House Environmental Justice Advisory Council I am now a part of. Not only environmental, but other needs in my community, including no adequate water and sewer, inadequate housing for the Arctic. These homes have no insulation on the floor. They're not well ventilated. There's no, when there's natural disasters in the lower 48, communities get immediate assistance. And we have communities in Alaska falling into the Bering Sea. They're not getting aid because they can't match the funds that some of these agents, state and federal agencies, they want communities to match these funds. When there's a crash in fisheries, fishermen get assistance for so that they can feed their families. We are now facing food security issues because the ice-dependent marine mammals my people depend on, including the walrus, our main food during our long winters. As the ice melts, our men have to go hunting further and these where it's more dangerous. We're facing food security. Our freezers are empty. Our elders and children are going hungry. These issues are so complex that it's going to take everybody, all of us in civil society, researchers, manufacturers, to make change protective of human health.
Great, thank you for that. We have one more question on HUVA, and then I think we might open it up to the other two speakers as well and have a panel discussion. This is kind of related to what you just said, but another attendee asks, what would be your recommendation for an effective community empowerment strategy to make effective action be more pronounced in the global regulatory conventions or in local regulations?
You know, we bring Arctic Indigenous. Was this toward me? This question toward me? Yes.
Yes.
We bring Arctic Indigenous Peoples delegation to the Stockholm Convention, an international treaty ratified by 182 countries, which the US is not even a party to. I've been to Geneva, Switzerland 5 times. I leave my home and travel 24 hours, 33 hours for 2 weeks at a time, and to testify and speak at the floor of the convention. We bring Arctic Indigenous people delegation. We are instrumental in helping to ban toxic chemicals globally that everybody on the planet benefits from. it is so important that we urge the U.S. to become a party to the convention, but others that have not. This is one mechanism. It's the only legally binding mechanism to address POPs. However, our national laws, state laws are very outdated and broken. We are very much involved. We have our own community-based participatory research project since 2000. We have had our own community-based project since then, looking at legacy chemicals and newer classic chemicals. The burden of proof have been put on us. So we are very much involved at the local level, state level, and national level. It is so important that everybody on this planet get involved to protect your children and future generations, the health and well-being of your people. It's important that anytime these issues are when the Arctic is, when we're talking about the Arctic, it's important that you bring people from the Arctic who are some of the most highly contaminated population on the planet to testify, to speak, where decisions are being made for us, usually miles and miles away. It's so important since we have the data of what we are finding in our lands and water and in the blood serum of our people.
Great. Well, thank you for that, and thank you for all your advocacy on behalf of everyone to protect people from harmful chemicals.
And I just want to make one quick comment. I was not able to download the app for this conference. I've had recent deaths in my family. Just a couple of days ago, I lost my sister. In my culture, your first cousins are your brothers and sisters. And within the past month, 3 of my relatives have passed away. So I was just now able to download the app so I can get back to those questions that were directed toward me.
Okay. Well, thank you for being here during such a difficult time for your family. So with that, I'd like to bring in the other 2 panelists. And to start off with, I was just curious if you had questions for each other. I'd love to have a discussion among the 3 of you and all the work that you've done.
I would like to thank Cora and John for all that you've done, because all the work that any researchers do benefit my community. So thank you for all the work that you've done. And I apologize, I'm raising a grandson as well who has been sick. So I will certainly go back and review your presentations and your slides. But I just want to take this opportunity to thank for the work that you have done.
Thank you, Vi. Okay, maybe I can have some comments and also questions. First of all, I can help you a little bit with this enhance question, because there is a global gradient, and your levels in the far north of the Arctic, they are significantly higher than the enhance levels. And so it's just the same all over the Arctic. And the other thing I want to, it's so interesting to see your slides, because you have exactly the same pictures from the military complex that we can see all over the Arctic, not only in Alaska, we can see it in Siberia, even in the north of Norway. So that is the mixture of having the global pollution that is simply going to the north, but also this endless barrels of contaminants just left by some military industry here. And that is really on the political level. And then the last comment now is that is a question I really don't know the answer on it. So maybe someone also in the audience can tell me what happens to the US EPA now after the change of president. Is there any change? Because I've been working with this for 30 years, and I've seen that the former president, he just almost eradicated the EPA. So where is the control system now? Where is the power now politically to continue the work? I would really hope that some of this, the control systems came back to give the hard evidence for the lawmakers, but I haven't seen it so far. So maybe someone can help me with that. Thank you.
We've exhausted all efforts with the EPA. I've met so many times. I've been doing this work going on 19 years. I've met with high officials. We brought a delegation to Washington, D.C. in 2009, and we got another funding recently to bring another delegation to Washington, D.C. We had meetings with high officials back to back in 2009 with EPA, with Army Corps of Engineer, who's responsible for the site characterization and remediation, with Pentagon, with our Congress people, with high officials. We've exhausted all efforts. They basically let the polluter off the hook. And recently there was an investigative report about EPA and corruption with the chemical corporations. You know, this is so unfortunate. They are not doing what they've been charged to do, which is to protect the environment, our lands and waters. And as I mentioned, you know, these health assessments are inconclusive by design. We are not the only community whose health has been harmed by the military. I believe they're the largest polluter on the planet. There's no funding. There needs to be investigations into EPA, into ATSDR, into the military. We are like losing lives. I used to be terrified to speak in front of groups, but I never knew there was so much injustice globally. And now I speak up because I'm a mother and grandmother. Help is not coming fast enough for my generation. I'm a cancer survivor. I'm lucky. Both my parents had cancer. My brother, though he's been cancer-free, is still struggling for his life. You know, these are health disparities never seen before in our people. And our nakabik so loaded with persistent organic pollutants at levels we shouldn't be eating it for excess cancer risk. So I am going to speak up and continue until, in Annie Aloha's words, I will fight until I melt.
It's very good to hear, because what I try to show you is also that the fluorinated compounds, that is like just the next chapter of something we have seen for a while. And the problematic things I showed is also where that some of the classical contaminants, they are nicely going down, even if they have long half-life, but the fluorinated compounds, they're going up. So maybe someone else can tell about how your this fluctuation to the north will also come into the human diet and the human body. We are very concerned about, for instance, like mercury, but also fluorinated. They are hormonal disruptors. So we have a long way to go, because now we have almost the evidence we need, but we also need to have to give the policymakers, the decision makers, this evidence. So that's really a tricky thing we need to look into now. Thank you.
Yes. You know, I don't have a degree, but I challenge myself to learn as much as I can, because this is, as I say, unethical. And not only for regulatory agencies that are failing us, but the polluters and the decision makers who are not making stringent enough policies to protect the most vulnerable.
Thank you.
Thank you for those comments. Reiner, did you want to ask a question?
No, I was trying to partially answer John's question. Some of our EPA colleagues on the research side are doing tremendous work to help understand, for example, where PFAS are coming from. But on the regulatory side, I think there is still a big silence in the US and nothing has changed that much. We've had action plan after action plan after action plan on PFAS, and so far there has been no change. The current administration seems to be pushing for something, but nothing has materialized. So we have to wait and see. I hope things are better in Canada and, of course, in Norway.
I had a question that I was hoping the panelists could address. We've seen that in Europe and in North America, the long-chain PFAS are no longer being manufactured for the most part, although there's still some production globally, and there's been this shift towards short chains and other alternatives, which generally seem not to bioaccumulate to the same degree. But I was interested to see in what Vi presented about PFPEA and PFHXA, which are short chain carboxylates being the most abundant in seals. So I was wondering, maybe Cora, you could start by talking about, as there's this shift to more short chains, what does that mean for the extent of transport to the Arctic, and what might we expect in terms of future exposures as we're still in this PFAS chapter, but it's sort of a new section as we're shifting the production.
Yeah, that's a great question. So we haven't, at least from my work with ice cores, we haven't seen any real evidence of that changing distribution. All of the PFAS continue to, basically all of them continue to increase with the exception of PFAS in the ice cores. So we haven't seen that impact of the sort of I guess, regulation that's been more local. And we think that's because the Arctic is a global sort of repository for these contaminants, and some countries continue to produce the longer chain materials. Now, transport through the ocean, I would expect we will see those effects coming, but they will be slower because the transport time through the ocean is slower.
Thanks for that. John, I didn't know if you had anything you wanted to add based on measurements in the AMAP program. Have you seen trends with short chains?
Yeah, what we see now in the recent studies that, of course, there is something coming up in our pooled samples from the north of Norway and Russia, gradually more short-chained. But what we saw in our last study here is this medium-chained, like the chlorinated paraffins, they're even increasing now. So there is something going on that we really need to look into, and of course. That I think that is the only way to solve this is a more strict regulatory policy, because the industry, they will do exactly what they are allowed to. They will not think like a warm-hearted the impact on the next generation. They will think what is allowed to through the laws, through the regulatory processes. So we need to take it from there. That's why I'm so curious about the EPA. Can they really do something better than they did in a formal period now? We have the EU system that is also producing regulations. Still, there's a lot of lobbying from the industry in Brussels, and I think there is something going on that we don't even know. So what we can do on our side is to have improved analytical quality, so we can even look at untargeted analysis, as Cora talked about, and we can do something that we still need to have hardcore evidence to give to the regulatory side and to the policymakers. Thank you.
Thanks for that thought, John, and that's a great segue to, I think, what's going to have to be the last question, since we're approaching 12:30 here. And just looking through the questions from Huva, there have been a couple questions about how can scientists best support communities. It sounds like one of your suggestions, John, is more measurements and using new methods, including non-targeted analysis, so that we can really demonstrate the extent of contamination. I was wondering what each of you might think of as other ways that scientists can do a better job of promoting effective change.
If I can just say one thing, it's just be generous to the media, to the public part of this here. Tell people how the situation is. And of course, all politicians, they're also afraid not to be re-elected. So give the public hard evidence, and then they might start to think about it, just to also for their own career. This is the future of the next generation. I used to call it, I want a new green generation. And of course, what I've heard now from Alaska today, this is really serious, and we need to address it. Also, we do it scientifically, but there is a political side we really need to address, and maybe use social media, all kinds of media to address our things now. Thank you.
I would agree with that, and I think public pressure is really the way to motivate regulators. And so the more we can do to motivate the public to feel strongly about this cause, the better off we will be. So talking to the media and being active on social media to raise awareness, this is what I do, and if others have better suggestions, I'm happy to implement them myself.
Thanks for that. And yeah, one thing that I've learned and have been told is to emphasize what you do know, and not just to talk about all the things that you don't know when you're speaking publicly, because there is quite a bit that we do know, and it's important to emphasize that. And Vi, I think I'll give you the last word for the scientists who are here. What's your final word here on what scientists can do to better support communities like yours?
Just, I have, as I mentioned, I've come to challenge myself to learn as much as I can. And because this is affecting the health and well-being of the most vulnerable on the planet, my people. And not only should you be working with the people, we should not just be subjects of research. Please involve the people you're doing research in. They should be coming up with the questions, what data to look at. And so that they can, it's a right to know issue, not only a right to know issue, but also so that they can bring their stories to the rooms where decisions are being made. Most people have no idea of what's happening in the Arctic. As my colleague John said here, this is a crisis. This is a climate crisis. And, there used to be 10,000 of us in 32 communities. Now we have 900 in my community, and our sister community, we have 800. 32 villages, now we have 2 left. And some of our elders say that there were 20,000 of us. So it's so important that when any research is done, that they are done with the local people you're working with, that they help design the research, that they are partners and not just subjects, and to use this data to make meaningful change at the local level, state level, national level, international level, because as a mother and grandmother, to me, I'm going to continue to say this. It's unethical. It's going to take all of us, because it profit over public health is and regrettable substitutions. So, our human right to health, human right to food and subsistence, human right to free and prior informed consent, and share my story, share what you learned from my colleagues, John and Cora. Bring it back to your workplaces and into your communities. Share our story and say that, that it's going to take all of us.
Well, I think that's a great note to end on. Thanks to all of our speakers today for a great discussion. Let's give them one more round of applause. All right, well, that concludes the session, and I'm going to hand the microphone back to Reiner.
Well, thank you very much, Laurel. That was, so I should point out the initial idea for an Arctic focus was from Philippe Grandjean, and then Laurel luckily fleshed it out. We had a fantastic ending to the deliberations of FLUOROS Global 2021. So big thanks to Laura. Quick question, there's lunch and it's sunny, but I have 10 minutes of content. You want to have a break and grab some lunch and be back? Or should I just keep going? Keep going? Okay, that was answered. All right, so let's see. I'm trying to play tag team with Alex behind the scenes. Let's see how well this goes. So, took us 2 years of planning, and then it was only 5 days of fun or stress, or whatever you want to call it. But I'm glad we managed to put in a global audience during COVID and have sessions that cover really the global extent of PFASs, the challenges we face, and some of the solutions that might be emerging. So the few things I realized, one is this Hoover thing apparently is really great, but it also has these little side things. And I was very distracted to realize I was only second on the Hoover leadership board. So it has to be bad. Problem that I was the top scorer as one of my students, so I threatened retaliatory action. But what I really wanted to get at is, so we tried something new. We tried to really have sessions across the globe and have recordings and restreaming and live streaming. And of course, sometimes it didn't always work, but of course there was always somebody there to help. And so this was our first, and we realized things were not going quite as we had hoped. So let's see if this works now.
Their proteins, their molecular transformation, their toxicity, property, for instance, endocrine disruptors, and they interfere with the gene expressions. We need more studies. So this symposium, I think as a part of the global FLUOROS conference, we will talk about the recent development in China and how we will join the international efforts, try to understand, try to control the pollution caused by PFAS. As so far we understand, PFAS are the most persistent, most polluted. You can find from every environmental samples, from the soil, from the water, from the sediment.
All right, so hopefully you can fix this behind the scenes. All right, so let's just wait and see. Maybe this just was a last minute idea, and he probably had to get some sleep. But let's see if he finds it.
Groups, namely the popular polyether PFPE and the short chain homology of long chain PFAS. For example, 62FTSA and F53B was replaced PFAS for metaplating.
It wasn't bad, but yeah. So what we're getting at is, he was talking, and of course, everybody's been on Zoom, and sometimes you're not quite sure what you're projecting. So after a few minutes, Dr. Ting Ruan came to help. His first try didn't quite work, so he came back, and then he fixed it. So things happen. So maybe we skip, and let's see if the next try actually works. All right, next try.
Biomarkers, and it's searchable. It contains summary statistics, and you can easily access them. You can select for the biomarker you are in, the chemical group you are interested in. You can select for the biomarker you are interested in, the age group you are interested in. So the data are.
So that was our live feed from Brussels. We thought that day everything was going to be smooth because there's nothing to do other than stream back from Brussels, and then Brussels disappeared. Luckily, because of things we could fix, it only took a few minutes to come up with an alternative solution. And then, of course, there was the classic. I was in the same session a little bit later. Let's see if Alex...
cohort in Sweden, from Sweden. So Christina, nice seeing you. Please, the floor is yours.
Thank you very much for inviting me to talk about our Ronneby PFAS research program. And I have to start to ask what you actually see on your screen.
And it was a very normal question, right? Because we all know what's on the screen, but it was funny because the screen, oops. The screen was actually working just fine. But it took a minute to realize that the screen was fine because nobody answered.
Slide, or do you see a double?
Yes, we see your slides perfectly.
Good.
Yeah.
So at least that day, the only trouble was always Zoom glitches, which is probably on the smaller side. And then we had this really nice talk that was so good. Oops. Let's see if you can pull this one up. That was from the...
In concentration with time, this follows an exponential decay relationship, which is consistent with the elutriation of fine particles from the soil. And so to conclude, experiments confirm the suitability of foam fractionation for the treatment of PFAS contaminated waters and fluoridization for the treatment of PFAS contaminated soils. This research was funded by the Australian Research Council, and I'd like to thank our industry partner, OPEC Systems Proprietary Limited, and the Australian Department of Defence for supply of materials. Thank you. My name is...
was Robert Neven's poster. And was
Thanks,
over.
Robert. Next one. I just always struggle to...
Robert Niven from the University of New South Wales in Canberra, Australia. I'm speaking on behalf of a team from UNSW, Macquarie University, Sydney, Australia. I'm going to talk about PFAS removal from contaminated waters and soils by foam fractionation and gas liquid.
All right, so take two is over now. Wait. Jochen is checking and...
Yeah, little hiccup, but we got there. And now we have to share screen. What would I do with a Sarah?
And it was actually, that was the third time when the same talk started being played. Anyhow, so it was a fantastic talk, obviously, so three chances. All right, the other thing that, and Jochen was very smart about this, you know, if something goes wrong, you always find somebody else who's at fault. So at the very introduction of his session, he started off with this.
As well as Zoom. This is Sarit, Dr. Sarit Casason, who is responsible for all the bugger-ups, and I'm responsible for all the things that's going to work out really well.
So clearly she was in charge of a lot, but it was a great session. It reminded me of, I think it might have been Anna Robach asking a question this morning. Sometimes we ask one or two questions, and then this person responding gets a little confused because it's very difficult to keep many things straight in your head when you're a little bit stressed and you have answer one and then what happened. So this is a great example of such a question. I think Ian Cousin was responding to a question, and just hear what he said about it. He's a very smart guy. It's just interesting.
In-person questions here in Providence.
Oh, this is.
I see Angie.
I was going to go.
Angela Slitt.
So I just wanted to, as an observation as a biomedical scientist, I think what's interesting is it doesn't seem like the industry is learning about the chemical structures and where the newer novel PFAS end up. And what I mean by that is that they're often acidic in nature. And so if they're acidic, their chances at a gut pH, they're not really going to be absorbed through permeability. They're going to be absorbed through transporters and other mechanisms. And so it's not kind of surprising that again and again, I know there's other mechanisms like FABPs and albumin that contribute to this, but in general, we're seeing the same patterns because in some ways they're all generating acids. Now, I know the neutral compounds are different. In our hands, when we measure them in cell-based permeability assays that are used in pharma, we do see that they're much more permeable than PFAS or PFOA. And so that will contribute to kind of, Ian's, I guess, Reiner's comment too. But one that sticks out at me that I didn't see was PFBS. And I wonder if it's because it kind of has some of the qualities that would make it not stick around and not stay in things. So it doesn't really bind to proteins well, whether it's albumin or other tissues. And even though it leverages transporters, it also leverages the same transporters in the kidneys to get out. So it gets in and it gets out, and it doesn't stick around. So I just wonder in some ways if maybe the engineering needs to kind of go in that way versus some of these molecules that are no matter what going to kind of do the same thing. They're high molecular weight, they're acidic, and they're just going to use transporters and stick around and deposit in tissues like the liver and the kidney where those transporter systems exist, but there's not really a good mechanism to get them out of the tissue.
Yeah, that's certainly what you say is very important. It's a lot to unpackage, but...
I love the Canadian understatement. There's a lot to unpackage. Yes, so excellent. Thank you, Angela. Perfect question of sorts. Yeah, there was something else that I found just funny. Let's see if Alex can actually pull these scenes up. But we're talking about Arctic, about wildlife, but for some reason monkeys came up. So let's see if we can get that one.
Of that. And there could be reasons for it, right? PFAS, say, creates a vulnerability. Monkeys with something that then makes makes the system, and that's some system within an organism, then more sensitive to another chemical.
But so clearly monkeys something up, but Mark Streiner followed up on this, and let's see if Alex can pull that clip now.
There's going to be hiccups. There was a paper on cynomolgus monkeys, and it was well known in that paper that when the PFAS was given to those monkeys, that essentially they all died in that study. And it's telling to look at a class of compounds where we have to remember that, persistence, bioaccumulative nature, and toxicology all contribute to why we work on these class of compounds.
So just for those that have kids, so he takes his children to work to tell them about stories where monkeys die in experiments. I mean, I don't know what kind of father he is, but my daughter would be having nightmares for weeks on end if I had this kind of experience. So I think he just turned somebody off science. But that's just my take. All right, the best question that nobody wanted to answer.
And then 2, how does the work you're doing help fill needed gaps in legal fields where there's a desperate need to be able to legally protect contaminated communities by showing links and showing exposures linked to harm, things like that. So I would love to hear each panelist's answer to those 2 questions, please.
Wow. All right. Eamon, go for it. Since you're on the different part of the screen. How does ECS help regulatory bodies?
You see he's sweating already.
I can tell you, this is a very tough question. It's not just a good question. It's a very tough question. I wish that someone else started before me.
All right, his answer was, please, somebody else step up. All right. I like this one because sometimes we get a question and we just don't know. But the question was very relevant.
Mashpee, we had 5 blue heron that had passed away in the Mashpee River, and they said that would be maybe 5 within 5 years. So I'm just wondering if there's anything that you have for testing, because they keep saying there's nothing in the water, or they can't find any reason why the heron are passing away. So I didn't know if there was anything that you had for testing for the waters, for the fish that are passing away, but more so the blue heron. Thank you.
Thank you for the question. I think we've recently collected some shellfish in collaboration with your team, and those results I don't think are complete yet, but we can certainly continue that conversation.
So the concern was about dying herons, and I think it's well known in ecology that often amateur observers, or people who have stewardship over the land, notice changes much, much quicker than scientists. But of course, the way we compartmentalize our science, we often don't know the answers, and I don't think Elsie knows the answer, but very smart response. All right, one more. This is a great answer, a question that Ian was asked, and let's just see.
Hi, Ian.
He was asked two questions.
Who ultimately gets to decide the what low concern is? I'm assuming it's a regulatory agency, but scientific community and industry going back and forth, at some point someone has to decide that. And then the second question I have is, do we know what the body burden is of background levels just in everybody's blood, and how that impacts low concern?
Yeah, the first question maybe is the easiest one, because I think it's not just regulators that are responsible. I mean, regulators I think, have a big responsibility in deciding whether fluoropolymers need to be regulated together with the PFAS, and whether polymers need to get more attention, because they've been traditionally been regulated separately from the low molecular weight organic substances, and so so I think regulators have a part, but I've also seen that consumers and product manufacturers are also playing a very big role. We had a seminar on PTFE cooking utensils, and in Sweden, a lot of the retailers who are making PTFE cooking utensils, who have made them historically, are now looking for alternatives and are actually trying to phase out PTFE cooking utensils. So that in Sweden, there's a retailer called Servia, and they've got a big campaign going right now to try and phase out PTFE cooking utensils. And also then consumers become more aware of the issues as well. Well, they can make consumer choices. So I think there's lots of different actors who can have an influence on phasing out fluoropolymers. So I think regulators, product manufacturers, and even the chemical manufacturers are making changes, at least. Even if they're not phasing out fluoropolymers, they are trying to make them in more responsible ways. So it's got. I agree that Gen-X is not really an improvement on PFOA, but the non-fluorinated processing aids, I think, are a better step. And they're trying to do more recycling, although I really doubt they'd say that. I really doubt whether they're going to really do much recycling. And then because it's just so expensive to do the recycling, like with lots of other plastics, to remove them from these complicated consumer products. Regarding the second question was about the levels in blood. What? Could you repeat that again? Because
So Ian just packed in 5 information pieces in his partial answer, and of course at this stage he did have the right to forget what the second question was. So I guess the lesson, if you ever ask a question, just ask 1, because clearly most of our brains just can't handle 2. But I thought it was a great answer. All right, this really, somebody deserves an award for this. It was the most efficient Q and answer session with a speaker that we had in 5 days. See if remediation session.
Okay. Well, and that brings us to the end of this remediation session. Just in time for Lutz joining us. Hi, Lutz. Good morning. I can't hear you yet. Early in the morning. Sorry, Lutz, there must have been some kind of miscommunication between organizers. Don't blame me. So everybody, have a great evening. Lutz, you have a super great morning in Sweden. Maybe we can catch up quickly afterwards and I summarize for you. And all the Queensland folks, we go out to a restaurant. Here is Lutz. Hi, Lutz.
Hi.
You had a good sleep?
Yeah. I think I thought it was a US time, but apparently not.
No, I'm sorry. I don't know how that got lost in translation. Maybe we should have spoken German with each other.
Yes.
All right, everybody. Have a wonderful evening. Jen and Carl and Lutz, thank you so much for your wonderful presentations. Really wonderful. My introduction was really lousy to you, but...
And then he muted himself. All right. So I think the way to avoid difficult questions is just to not show up in time. All right, and now there's only 2 highlights, and this one was very nice. This was when we had that little loss of communication to Brussels. Philippe Grandjean took over, and he had to introduce the next speaker.
Okay, I hope I'm on. It's very nice to be part of this experimental FLUOROS conference. And since we all work on experiments, having this going on at a scientific meeting is really the peak experience for, at least for me. So I'm very happy to introduce the first speaker who's not present, but who recorded this a while ago. I think that he is introducing himself. Alex or Jared, can you put on the first video?
See, doesn't he want to introduce the next speaker?
The video is named Grandjean, I believe.
Good afternoon, or good morning, as the case may be. I'm happy to join you, although it's just going to be via my computer screen.
All right, and I think the best one is the last one. It's the best. Thank you for the speakers. So the last clip, Alex, please.
20-minute presentation. I want to thank the speakers and the organizers, and as well as Steep Funding for this meeting. I really hope you enjoy this session, and I look forward to a lively discussion during the question period. Thanks.
Okay, great. That was a very good presentation, Emila de Silva.
All right, so with that, it was a great pleasure chairing this conference. Thank you all for participating. I think we had stimulating science. We had a lot of science from across the globe. With that, this is now officially over. Thank you, everybody. There's still food in the back, and might see you soon at some other meeting. Thank you very much. Bye-bye.